So just hit full
Okay.
screen. Hit full screen in your PPT, in your PowerPoint.
Yeah, that I'm doing. But even
Yeah, we can see your
okay.
slides now. Yeah.
So I'm just seeing my screen. Even I can't see Zoom, I can't see time. Nothing I can see.
Yeah, because that
So
is
is it?
that is open in a screen which is not the active application, right? You are in PowerPoint, right? But
Yes.
but Zoom camera is if you want you can switch on your camera.
Yeah, I can switch on my camera. Perfect. That is
We can
working.
see you and we can see your slides.
Perfect. No problem. So I think this is okay.
Do you want to take a couple-minute break or you are ready to start?
I think we should start and I think we should finish by time. Otherwise, we'll miss lunch. No, it's
Okay.
already. So what is the target time, Ashish? Like roughly when I have to finish? By 12, I have to, no, by 1, I have to finish and then maybe 10 minutes go for questions. Means
Yeah.
I'm ready that if the questions are in between. So I really like to have questions at any moment, at any slide, because I think if the students want it more interactive, that will be really helpful. If not, then anyway, I can keep talking, and probably it will work.
Yeah.
So my
I
point is,
think
stop me at any point. I don't have any issue with that, and I really think that whatever I'm talking, it's at least 50% of that should be understood by others.
so.
Okay.
Attendees, this is another call. Please feel free to
No, I
interrupt,
think
ask questions.
probably there. Don't be shy. I think it's just like we really hope that you know the way we are doing our research or what thought is in our minds to do it. So that is more interesting to understand from person who is doing. So I think it's like not about the questions. You can ask any question that why it is done by this way, why not this way. At least I may have some answer for you. So because with all my experience since last yesterday, I am feeling that either you are getting everything or you are getting nothing. That is a physicist way to say that. That's how it's state 0 or 1. Anyway, so let us start. Uh,
I'll
Okay,
quickly
Ashish?
introduce you. No, I'll quickly
Sure,
introduce
sure,
you
sure.
and then
Perfect.
we can start. Yeah, just
Sure. That either you are getting everything or you are getting nothing. That is a physicist way to say that. That's how it's state 0 or 1. Anyway, so let us start.
yeah, I'll quickly introduce you.
Okay, Ashish?
No, I'll quickly introduce
Sure, sure.
and then we can start. Yeah. So some technique. Okay, 1 second. I am just. I think there was
Yeah,
some. Yeah. So Professor
yeah.
Garima received her PhD in physics from BHU, Banaras Hindu University, Varanasi. She did her postdoc at Weizmann Institute of Science, Rehovot, Israel. She was also a post. Okay, 1 second. I am just. I think there was some. Yeah. So Professor Garima received her PhD in physics from BHU, Banaras Some Hindu University, stream is Varanasi. Playing somewhere. She did her postdoc at
Yes.
Weizmann Institute of
Ashish? Ashish?
Yeah, ma'am. I think you can start. There was some duplicate stream playing somewhere, but should be okay now. Can you
The
hear me?
slide is visible.
No, slide is not visible.
Ashish? No, I think it's repeating, it's echoing, Ashish. There is some problem.
Duplicate stream playing somewhere, but should be okay. Ma'am, give me 2 minutes. I'll try
Should
to
we
find.
should we join again or something?
Just give me 2 minutes. Give me 2 minutes. I'm
No,
trying to.
sure.
Ma'am, it's fixed now. You can go ahead. Sorry for the inconvenience. You can share your screen and start. Yeah.
Is it okay? Is it visible, Ashish?
Yeah, yeah.
Okay, perfect. So
Sorry.
today I'm going to talk about DNA stability through coarse grained simulation. And as I assume there are some students, so I think I would like to first emphasize on simulations, which could be computer simulation or simulations of different kind, coarse graining. And then we will slowly take the topic of DNA stability in some context, some biological context. So let us start. So this is very famous. So I repeat this slide everywhere. And the reason for this slide is very simple. That if you see, this is, I have taken from this appsschools.com. This is like a comic site. Earlier it was supposed to be. Now they are not publishing anymore. But let us see this picture. And the first question will be what we see. And they're in the audience also. That's why I say ask question, that when we say what we see, there must be, at least at this moment of time, I can ask some of you, if you are there, what we see. Can anyone tell me what we see? Anyone? At least here you shouldn't be hesitant, no? Whatever you see. You don't want to answer this also? No answer? Wow, this is going to be really tough audience. Nobody want to say what they see?
There are a lot of things we can see in this picture, ma'am, like trees, like a rabbit eating a carrot.
Perfect. This is what you're seeing. And someone else is seeing something different here, except rabbit, trees, and this flying birds, something.
Sun. I don't know.
Sun, perfect, perfect. Sun is also there. And likewise, someone will see river, someone will some. And the important point to note here, and for this whole 1.5 hour talk, is really that whatever you see, just put in there and say something there for your understanding. So let us see how a scientist will see this picture. So if this, yes. So you see, this is how a scientist will view. Or mostly I can say a physicist viewpoint. Mostly there are equations, some chemistry, some biology sitting here. Okay, so this is another viewpoint, that if you want to see what's going on, why there is a light, you will ask these questions, no? That, okay, there are the Maxwell equation. If someone of you know Maxwell equation tells you about this EM wave, light is one of the EM wave. And then also there's, if you go to the chemist, they will see, okay, let us write some equations and figure out that how these processes are working. So look at these equations. There are many ways to see the same thing. And trust me, everyone's viewpoint is equally important. Doesn't matter whether you are a physicist, chemist, biologist, or for that matter, not a scientist. Then also you should appreciate your thought and you should ask the questions. So this is just to keep you into the talk, that whatever viewpoint you got, just tell it and ask the questions. Next important thing is that if you are doing science, because we are, Ashish is not working. Why is there? It's my end. Okay, so let us say that we are a scientist, say for example, even a non-scientist. So then in all these cases, we have some questions of interest. So we see something and we try to understand some aspect of this. And in that case, if we are taking biology, physics, chemistry, or any branch, so then we'll ask some questions. And that some questions will be a real system, which you can design, which you can probe. So on that system, you can ask your question of interest. It could be on viruses, it could be on DNA. If you're a biologist, protein, their interaction. If you're a physicist, you can do again the same exercise. So the point is you design a system, perform the experiment, and you have experimental results. This is one way, a standard way of doing some research. The other possibility is that, okay, you have a real system and you don't want to do the experiment. You simply have a model system. You can make a model for a system How close model you develop, that is important. But once your model is developed, let us say you guess the model, and then you make some theoretical hypothesis and make some predictions. Similarly, if you can make the prediction, you can match with the experimental result. And other way is to model the system, put the system with some sort of sim computer and get your simulation result. This simulation can be of any type. You even can write an equation and then can simulate that and find out the results. Again, keep in mind, we are answering the question of our interest by using different means. One mean is just simply doing the experiment, we got the result. And other mean is more theoretical viewpoint, where we have this pathway. Either use pen or pencil to get the theoretical predictions, or use your computer. But the end goal is to understand this question of interest. And these three outputs are not independent, because you know that you have experimental result, okay? And those experiment results must have some underlying mechanism, some behavior that simulation and the theoretical prediction can help you to understand it more. And let us say that your the theoretical results are different from the experiment. Then you try to do something here in the modeling of the system. You change some sort of modeling so that you can get the results which are closer to the experiment. Okay, once you got it, then you are in the upper hand, because then each time you don't have to perform the experiment. You can use this modeling, whatever the model you have developed, and keep doing this and get the output. So why we need simulation? The simple reason is this, okay? So all of you who are doing experiment, at least they will agree with me more that it is really difficult to perform experiments. So even biologists, it's really hard to grow the culture and keep them. Even moreover, the problem I see the experiments in biology, and I really appreciate their hard work, that they take really long time, and keeping the whole environment, the whole system insensitive of the environment is crucial. So each time performing the experiment is a bit tough, expensive, of course. And in some cases, I believe that you have too many parameters to control, and you don't have that, you don't know that which parameters is controlling what. This is the some sort of limitations of the experiment, I say. Of course, it's not like the nothing is foolproof, but this is limitations, and which is these days actually overcome by these computer simulation. Theory I'm not putting, because most of the theoretical result you can't solve many problems with this pen and pencil. You can do some, but not most of them. So there comes the experiment, where you do the experiment, but in computer. And once your modeling is perfect, when your model is developed very nicely, then you can simply use those models, predict something for experiment, and moreover, you can complete the whole experimental and theoretical part. You can explain the observation, why something is happening in the experiment, what is the fundamentals behind that. So that's why we are interested in simulations. And which type of simulations? So simulation can be at different levels, okay? So simulations of the system, for example, can be at electronic level, electronic wave function level. Those of you who are not aware of this, this is simply like you write the Schrodinger equation, that is for electron. This is just a Newton's equation of motion for electron. Keep in mind, equation of motion for electrons. You solve those equations, and this is called density functional theory. But again, the restriction is that you can't do it for many particles, many electron system, or if you can't go more than 10 or 20 atoms. Then you keep doing some more coarse graining. You go from this electronic level wave functions to atom level simulation, that is called ab initio molecular dynamics, where you mostly have these softwares like Umber, Charm, GROMACS. So there they take the details of each atom and they simulate the problem, whatever the problem. So we always have some problem in our hand, and based on the problem of interest, we do these modeling. Then, of course, we have a classical molecular dynamics and coarse graining. Coarse graining is the reason where I work. And in today's talk, probably we will see something in this reason of coarse graining. Now, the important thing is in these simulations, what is this MD? So molecular dynamics simulation is the where you solve the equation of motion, and we know what is the trajectory of the system, how it is evolving with the time. And the K property is like Feynman said, that at the bottom level, even biology, take any system, consider anything. They're not frozen. They are always in dynamic behavior. They're wiggly motion. So all these atoms which you think, They are continuously in the motion. And biology is best to study this because, all the biologists happen the temperature and they do numerous amount of functions. Biology is full of functions and complexity. So it is really nice to incorporate these simulations and study the biological system. Of course, you can do this simulation to any system, not only to biologists. So even the people use this Umber and these some softwares to study the material also. But that is not the problem. The problem is that where we want to apply it. You can apply it anywhere, all these molecular dynamic simulations. Like Sachi was telling in her talk, that everybody is using molecular dynamics because they want to know about these wiggly motion, how these atoms which are connected with each other, they are moving. So what is the fundamental principle? Fundamental principle is again, okay, so fundamental principle is very simple. You have to generate as many as conformations you can. And it has been saying in the physics that if you have enough number of conformations, like if you had the talk from Anand yesterday, he was also mentioning that sample the whole space. If you can sample the whole space, probably you can know everything about the system of your interest. And this comes under the name with the Boltzmann sampling or Boltzmann distribution. You have heard from Anand. And once you have this enough amount of conformations, you can simply, from these conformation, you can get the information what you want. Okay. So this is just to tell you that how the simulation works. Simulation can work at different levels. And what is a fundamental equation in these simulations is nothing but the Newton's equation of motion. So where you know that there are forces, so think about your atoms. Now think the whole system you have, combination of atoms, you have a forces, and if you are under the forces, these atoms will gain some acceleration depending on their mass. This is that's all. Nothing else is there in these simulations. Just one line equation. Of course, the solving this, integrating this, that is a different story. But the important thing is sitting in this force. How you design this force. And this force is nothing but gradient of potential. So this is a very standard form. You have potential because you design your system in terms of energy. Some sort of interaction you have to tell. So first important question, if any of you is using this Umber, Gromax, some sort of software, did you question yourself, for example, that all the way you have learned this, that you have to solve an equation of motion F equal to ma. But F equal to ma is a classical equation. Did you ask this question that how I can use a classical equation to study a atomic level problem, for example, the Umber? Did you ask this question? Any one of you using the Umber or some software? Anyone? Yes, no? No? So let us say that you don't, none of you used it. So this talk might be more useful in that case. So the point here is that when you write these potential in Umber, okay, so they are not depending on the exact. So even if you have some memory from the first talk of the yesterday meeting, from Lipi, that she mentioned about some bonded, non-bonded interaction, and she plugged in this interaction in this potential. And then once you have this potential gradient, you get the forces, and then you can plug in, integrate the equation of motion. The important part here is this potential is a empirical potential. It's nothing to do with the classical and quantum. Somehow the people design the whole exercise behind developing this software that you optimize your potential in such a manner which can be used to predict some behaviors of your system of interest. For example, Umber can use very nicely DNA, Gromax, protein, unless you have your homemade code. So the whole purpose is to develop an empirical potential based on the experimental observation. So goal is that you develop a potential, then you can make some observations, match these observations with the experiments. Once it is okay, then you are in very good condition and you can use this model potential to study something new which you couldn't get in the experiment. That's the whole idea. Now, this is the overall technique which we are going to use in this talk. This is just writing this equation of motion. And every time I'll tell you some problem, I'll tell you about this specific potential, that why this potential is very important. And if you tune this potential, how can you change the properties? And did you get some new information from here? So let us start with a problem. So I will give you a simple problem. So you all know what is DNA. Okay. DNA, if you are a biologist, for you, DNA is really very important. For us also, for our functioning also, DNA is very important. But we also see DNA as a very, very long polymer. It's really a polymer because it is made up of some units. Of course, there are 4 different type of nucleotides. So there are 4 monomers which can arrange in different sequences to give you gene information. But moreover, if you take a DNA from one of your cell, can you tell me what is the size of the DNA if you bring it from one of the cell? Anyone is biologist there, sitting there? Any biologist?
Self-taught, 3 billion base pairs.
3 billion base pairs. So what is the length? Roughly 3 billion. So for example, each base pair is roughly of the order of few angstrom. The distance 3.5, 6 point something in number, 3.4 nanometer, the distance between 2 bases. Is it okay, Ashish? So if we can simply do a small multiplication, how long our DNA will be? So 6 in 10 is for 9 nucleotide roughly. And each of these is 3.4 angstrom, roughly 0.34 nanometer. And just multiply this. This number is 2 meter. So can anyone tell me what is the average size of a human? Average size? This is from one cell. What is the average size of a human? Not more than 2 meter for sure. No way. Roughly 1.5 meter or so, unless we are exceptional. So think just one cell DNA, you bring them out and you can see the distance up to 2 meter. And cell size is hardly 10 micrometer. At least you should agree with this. And if you don't like these numbers, I can give you very nice numbers. Let us see that numbers and just let us rescale this number. How I'll rescale? I can simply multiply these both numbers with 10 raised power 8. That's all I'm going to do. Because we are used to of these scales, the centimeter, meter scale. So you know what this 2 meter will correspond if we simply multiply with 10 raised power 8? It's 10 raised power 5 kilometer distance from Earth to Moon. This is our Earth, this is Moon. So this much distance, and where we have to fit it? We have to fit it in this region. So can you see the cursor? So our Ashoka, I don't know how many of you from Ashoka, but this is our campus, a small campus. So we have to fit this distance from Earth to Moon in this small region, if you can see my cursor, where this is Ashoka sitting here. Is it interesting? So it is definitely interesting for biologists, but it is more interesting for us also. Us, when I'm saying that I really think that this compaction is huge. So what gives this compaction? Why we can do? Of course, there can be many complicated mechanisms, much more complicated, which I can think. But on the simple ground, what first thing I can think about? So as a physicist, we always have some fascination with the biology because we work with polymers. And the biological systems are really very attracting to us. So we try to answer them with our understanding. And if you look at these DNA by this chemographs, so even these structures, what I talk you that they will be roughly a stretch of 2 meters. They're not just stretched. They are having a conformation which are more like a sort of random. They're not just stretched, like you keep a thread with you and just leave the thread. Do you think it will remain stretched? No, it will not be stretched, but it will have some size. So what is that size? That size is like, if you look at the chemograph, you can see there are many bends here and there. However, the conformation is not like predicted easily, but there is some sort of randomness involved. And what this randomness? So when we say randomness is not as if random. It also gives you some information. So what a randomness can give you? So simplest model of polymer is a random walk. Can I use a random walk to model DNA at first glance? And if I do, what consequences I will get? Let us see these consequences first. So random walk is nothing. Just take a coin. All of you, just take a coin and say that, okay, if I am allowed to move on a one-dimensional line, let us say I have to move only on a road in either in left or on right. That's the only thing you can do. You can't jump up or you can't go below the earth, of course. So you can't go below the surface. So you can only go either in the left direction or right direction. You have a coin. Coin have a head and tail, and you know that your coin is not biased. So if you flip the coin, can you tell me that what is the possibility that on an average after 100 flippings, you will have a movement? Any one of you can tell me something? Should I ask this question or not? Anyone?
Ma'am,
No?
I don't know if the audience may be familiar with random walks.
No, so that's what I'm saying. So let us say not random walk. No, let us say that we have a coin. That's why, yeah, that's the whole emphasis. My point is we have a coin. And I'm standing, let us wherever you're standing, flip a coin. What is the probability? What is the thing that I will move left? How many times? Is it possible that I flip the coin, then only I'll decide my move? So can I keep moving in left? Is it possible? Forget about random walk. Flipping, let us do the coin flipping. Is it okay or not?
So it will be a series of like conditional probabilities, right? Like if you
no, it's just random. I'm saying I have a coin. If it is head, I'll move left. If it is tail, I'll move right. That's the only information I have. Nothing else I'm giving you.
If you move left, then there is another.
Then I again
do another.
Then again,
flip,
again.
Right.
No, then again I'll flip the coin and I'll see whether I can move left or right. So what is the consequences? Do you think that, or do we think that we can keep going and after some time we can simply go to the a net 1 kilometer distance just by flipping coin? No, you can't, because it is possible that half the time we'll get head, half the time we'll get the tail. Is it okay or not? Yes or no? Yes or no?
Yes.
That is a much more simpler version. So next time, I think this whole meeting, I really wanted to tell you that do this experiment. It's a really very nice experiment that you flip it and see where you're landing, which position you're landing. And take my point that most of the cases, if you do this experiment enough number of times, your net displacement is 0. You will not move from the position you started. And if you do this, the next thing, you'll see that this R is nothing but the displacement, some sort of displacement. You see this displacement is 0. However, if you do the square of this is just a mathematical input. So if you do this R squared thing, then you get some finite contribution, not 0. Because you are having left square is some positive number. It will not cancel out, left will not cancel out with the right. So you will get some finite value. And that finite value is telling you about this displacement. And the same thing holds for the DNA or any polymer which you can think about until unless you distinguish about the atomic details, which we'll do, but at later stage. So at first glance, if you do this exercise, you see that this DNA, which was 2 meters long, by doing this simple random walk thing, is somehow compacted in 25 micrometer. So you see, we started with 2 meters. And now I'm saying that even a simple polymer physics principle will tell you that this is not 2 meters. You can compact the whole thing in 25 micrometer, but still your cell size is 10 micrometer. So then biology comes in the picture very nicely that you have, this is not the end of the story. We know that there are proteins, nucleosomes, which are the histones which can form the nucleosome and then further compaction. So these compactions are there. But this is the first step is you can, even if you don't have the histone, then also you will not get the 2 meters. You always will get something around 25 micrometers. Just to add on this, you keep a thread with you. Whatever thread you want, just try to keep it stretched, holding it far apart. You can't do this. It will always come back. And this is the very important term called entropy in physics. So by doing the stretching, you are reducing the entropy. But if you are bringing them close, the size you're reducing, you get more entropy. And entropy is also equally important in biology. So with this compaction, you are happy, and there are further compaction by the histone. You can make at least some further compaction, and this 2 meter long DNA could fit in the cell. Now, the next question for, at least for if biologists are sitting there, what about the bacteria? Do you think that we need more number of bases in the bacteria or less? At least this much you should say. Anyone? No biologist is sitting.
Relative to humans, do you mean?
So for example, I'm asked, so do we need more number of nucleotides for bacteria or less?
Compared to what? Compared to humans?
Compared to humans, yeah, of course, 6 × 10⁹.
Yeah, it will be less.
Yeah, so Ashish, except you, can someone tell me why?
Because intuitively we feel, okay.
No, of course, no, that's how that we can keep talking. But the point is that, is there someone want to jump in? Like everybody's viewpoint is important? No? None of you?
So anyway, bacteria are the length scale of bacteria is much smaller than a human being, and the complication level of those organisms are also much lesser. So one would expect that
Perfect.
the number of
Yeah, I agree.
bases.
I agree. So this is, at least this makes sense, no? That bacteria are not that complicated like us. They may be, but at least they don't have to perform as many functions as a human should be. So even the less number of, because in the very beginning, we mentioned that it needs to somehow have genes or some functionality. So we don't need that many informations. Now, but the question is again, so in the bacteria, you have 1 millimeter long DNA. In humans, it's 2 millimeter in one cell, now 1 millimeter. What is the size of bacteria? On the same ground, I can say that even if you do all these random walk thing, then also you will see this compaction 5 micrometer is still really big because of the size of the bacteria. And how many of you will agree with me, at least, that these bacterial DNAs are much more nicer because they don't need this nucleus. Do they need nucleus? Are they inside the nucleus? Yes or no? No one?
No, bacteria don't have nucleus.
Okay, it doesn't have nucleus. So my point is that it doesn't have nucleus. And do they have histones? Like sort of protein-like histones? No? Answer? Yes? Okay. So my point is that I'm not biologist like you, but I have questions from biology, and I really think that all of us to work together, at least to ask the questions. So because I can do these things very easily, but the point is, until unless you come in and say that, this is the bacteria, I'm saying this is a bacteria and this is a DNA, you should say something. That, okay, histone is there or not, some sort of histone is there or not. So if not, then how the compaction happen? Or it is not interesting at all. Why should we bother about the compaction? This might be another answer. So earlier view of the bacterial DNA was like the whole DNA will be simply like a soup like structure. Long back, this was the whole hypothesis, that this, because there is no nucleus, it was simply floating around. But modern research are actually telling with these fancy experiments that these DNAs are not like soup. They're not like simply distributed. They are in the very small region. This is non-membrane region, but they are called nucleoid. And in these nucleoid, the question again is that if there is no histone, what gives rise this nucleoid? Why this compaction? So there are some very nice hypotheses, and we try to check those. So my question is starting like how this compaction happens. And if you look this picture of bacteria, you can at least propose some simple minded mechanism. The first mechanism, the DNA has a charged backbone. It has a phosphate in the backbone. Okay, so this is negatively charged. So if you have just charges, your high school level physics, even that will work. So if you have just positive charge on the backbone, what type of things you can build? Do you think about the compaction? Do you think that this charge, let us say we have charge sitting together, a backbone, will they compact? No, they will not compact because charge repulsion is there. So one possible mechanism could be that maybe inside the cell, probably these charges on the backbone could be neutralized. And if they are neutralized, at least you can still hope for some sort of compaction. So one possible mechanism was, this is just a hypothesis, And sometimes there are some experiments which supported it, that okay, the backbone charge neutralization is one possible way for compaction. The other possible way is that it might be that not all the DNA which we are talking about, they're not alone. They're not sitting alone inside the cell. The cell has a very crowded environment. There are many other things more than the DNA. And probably those environments are also helping in this nucleoid formation. So what those environments can do, for example, probably DNA doesn't want to like to be very close, like form a nucleoid because of something. The charge or repulsion or screening, they may come compact. But it might also happen that they don't have a space. Where else they will go? So they have to be there. And think about a situation that we are in a classroom and there are many people coming in. So initially, all the chairs and the tables are distributed. So now if there are many students coming in, what the best way I can ask them to sit? I can say, okay, fine, you know what, let us stack chairs and table together and make more space. But do you think that the tables and chairs are interacting? No, they are not. They are just creating some sort of space by stacking together so that other students can come and find some place, even to stand up. So this type of things can happen in biology, that because of the presence of these other molecules, the RNA polymerase, RNA, there are many things in the cell which can actually help the DNA to compact, bacterial DNA to compact, because there is not significant amount of histone. Even the experimental study suggested that if you want the compaction from this nuclear associated proteins, NAPs, you need huge concentration. That is not possible in vivo. In vitro, you can do. Of course, this experiment hasn't performed in vitro, and they have seen that if you keep increasing the concentration of the NAPs, you can make them compact. But that huge compaction is not there in vivo. So this possibility is also ruled out. And other thing is that there was another experiment in 2010 where they put this BSA protein. This is a very high charge protein. High charge, but opposite charge. The negative charge on the protein and the negative charge on the DNA. So what happens that when you have two negative charges, keep in mind, DNA is floating around and then we are putting a negatively charged protein. So what will happen? It will feel the repulsion because of the presence of other negative charges. That will also give you the compaction. This is called segregative phase separation. And this is a very nice paper where you see, this is experimental paper, where by increasing the concentration of BSA, they found that the DNA get compacted. This dashed line is just the original DNA, which is here. But if you put the BSA in more amount, the whole DNA get compacted. So far, is it okay? So I'm just telling you the motivation for a biological problem, the biological problem, which I feel interesting, that what could be the possible reason? And of course, we should go for the simulations, and then we'll see that, okay, what gives rise this compaction? And if we do this by simple-minded approach, okay, one more question comes that if there is a compaction in DNA, which is in terms of nucleotide in bacteria, does this collapsed DNA melt? Melting means simply some sort of opening. Okay, so if it's simply like, let us assume that it form a very different type of structure. So is it easy to access the genes inside or the sequences inside? Means let us make a compaction. We can make a compaction by some means. And then the question I'm asking that how to access the sequence in the DNA? Is it okay? Any questions? Do we need to access the sequence or not? Or we can work without accessing the sequence? Is it anybody?
Yes, I think
Yes,
one
like
needs to.
Yeah, we have to access the sequence. But can you tell me, I'll give you a random example. Let us say that somehow I gave you a DNA, which is very randomly packed. Means it is packed, but very randomly packed. Will it be easy to get the access of the sequence easily for protein? What will be easy to get the access? Is it easy that it has some sort of a structure, or it is easy that does have a very random structure or buried structure of sequences, and DNA the protein should go and access the sequence? Which one is the easy one, or what one will hope? Any hope here?
The easy access will be for those region of the DNA which are like a little bit of like out of those random folds, like
Yes,
easily
random
accessible.
folds, easily accessible. Perfect. So that is the whole thing. Even the biology shouldn't think that everything is very badly compacted and you should simply go there, access. This will be tough. So the next question will be, is this compaction will melt or the DNA will melt and give the access of these sequences to other proteins for proper functioning? Because we all believe that even bacteria will function in the similar manner. For example, the strand has to separate, then the sequences needs to be accessed. So when I say the word melt, it simply means the separation, some sort of separation between two strands. That's all. So now, can we model this system? So we know some possible mechanism that, okay, there are some possible mechanisms. We are not very sure which is exactly the mechanism. But the question we are asking that, can we get a structured structure, and then can we get the melting without getting some fuzzy phases in between? So to model this, we have so like I said in the very beginning, for all these questions, whatever questions from biology is coming, we are utilizing the tool from computer simulation. And the simple thing is sitting here is this U is simple energy functions. And then I integrate the equation of motion and I'll get my solution. So for me, the problem is that, do we think the bacteria in the DNA get compact? Yes, because there is experimental observation. There is a nucleoid reason. So they must get compacted. And the second question is, will this compacted DNA in bacteria can melt? These are the two questions for us now. Once we can get this answer, then we can go for the validation and the future prediction. So we have to validate these questions. So while course of the study, we'll see that can we validate some of the observations. So we are doing simulations and some observations we can reproduce the experimental observation. And if not, then can we suggest some experimentalists that can you perform these experiments? These are the standard way. So to do this, we use the coarse-grained modeling. I think the last speaker also talks something about the coarse-grained modeling. So you can model this system at different level. But the important point is that what you want from the model. Or in principle, I always say this, that it's a limitation, technical limitation. You can definitely use a big, nice software and can run whole thing, but there is a limitation involved. So can you have that much amount of supercomputer access? Or if not, that what best you can do? How can you bring this several parameter variable in few variable parameter? Like yesterday, Anand was doing. So principal component analysis. Okay, his topic was different, but the whole idea was that bring this many variable problem to few variable problem. So can we do something similar in the DNA? So of course, for DNA, if you look at the literature, there are many nice simulations. And all atom simulations, like I said, where you have information of each of these atoms, carbon, sugar, phosphate, everything you want, you can plug in. The other level is residue level of coarse graining. There is ultra coarse graining also, where you can have like some sort of polymeric behavior. And similarly, one can do for protein also, like Sachi was saying that you can have the coarse grained modeling for protein. But when you keep in mind, when you're designing a problem, you should know that what you are interested in. Whether you're interested in really the specificity, that you have to know the system about each residue level or each nucleotide level, or you're really interested in studying the large wavelength behavior. Large wavelength behavior means a behavior which is not, of course, it will depend on the specificity, but not that much. Okay, so in that sense, what type of modeling you want to use? Because this whole constraint is about time and the length. So if you have very, very large length of DNA and whether you want to study a nucleotide level of details, then probably you will stuck in the time. You may not get that much supercomputer time or something. So you have to optimize your problems. So what we did a very simple minded approach. We used it. And like I said, that every problem I'll discuss with you, I'll tell you that what is the fundamental, how I was taking the interaction potential. Because keep in mind, in all this simulation, the important part is this interaction potential, because the integrator anyway you can bring from anywhere. So you can use standard integrators. And once you do the integration of the equation of motion, then you get the trajectories and then you can do rest of the analysis. So Can any one of you tell me that if you have a free, let us say that in DNA, we are having the nucleotides, okay? And there is a sugar phosphate backbone. So what is the first thing you should think about the backbone? Backbone means what? What is a backbone? Can it be possible that you can keep cutting the backbones? Like if I cut the backbone, let us say I separated the sugar phosphate by some way, like there must be some nice enzymes which can cut it. So this cutting will...
Backbone refers to like the scaffold to which other things attached.
Yeah, so yeah, of course, means in principle, if you want a DNA, my point is that when we have a DNA, what is the fundamental characteristic of the DNA? So if I keep it cutting, let us say like I have many, somehow I have luxury to get many enzymes which can cut the sugar phosphate backbone. Then will it be DNA or simply the nucleotide floating around? The question is clear or not? Will it be DNA?
Sure.
Ashish, no. That okay. So maybe it's a way to say, for example, you have a DNA. Okay, so we have a double helical structure. Agree? Now let us say that, for example, there must be some cutting enzymes, no, in the biology. Must be who can simply make the cuts.
Cleave. Yeah.
So they can make the cuts. So my point is, if I have abundant number of these cutting enzymes and you gave me a DNA, I put them together and they cut all these backbone phosphate sugar links, then will it be DNA?
Ma'am, do you mean the the phosphodiester backbone, or like you are asking?
Yeah, phosphodiester backbone. Exactly, that's the word. Perfect.
Yeah.
So this backbone. Yes, Bishal.
No, yeah, there are enzymes, yeah.
So if you can cut it, so my point is then it will be no more the DNA backbone. It will be almost gone. So first thing in the simulation, we should ensure that the backbone remains there. And don't bother about this expression. This expression has nothing except telling you that what is a fundamental requirement for my DNA. The fundamental requirement is that there must be two strands. Okay, the two strands. So let us say this is one strand. If you can see my cursor, let us say this is one strand. And the strand is not like a freely moving phosphate sugar. They are connected. So this first term will just take care of these connections. Then there is another thing which we want in this strand, that two strands are also bound by the base pairing. You know, this helical structure where the AT and JC base pair forms. So you need some sort of attraction. AT base pair involves 2 hydrogen bond, GC base pair involves 3 hydrogen bond. I know that you guys are well expert in these informations. So, but the whole important point is, if I can have these AT and GC base pairs, they can form a bound structure. So this last terms, like this, some expression, I don't think you should bother. If you know this term, that's fine. But if you don't know this term is just to ensure that you can keep these base pairing. There are two strands A and B. You can somehow simply bring them together and say that you be bounded. Okay. And another important potential which we are trying to look for compaction. See, the problem is still we are with the bacterial DNA compaction, whether we can make them compact or not. And there are many hypotheses. So one present modeling we are doing, okay, let us do some simple-minded approach. And in that, I will give some sort of interaction. And that interaction is very simple. That, okay, each of these red dotted line you can see, it tells that these beads can form a fold. So the beads can form a fold. Wherever the red dashed line, can you see my cursor? So they will form a fold. So if I give you this bead, polymeric bead, which are connected, and I allow these type of folds, can you tell me what type of structure this will take? Just first term, first figure. So it will form a ladder. Take a wire, fold it. That's the only thing it will give you. A ladder formation by this force field or energy, whatever you want to say in the simulation. And similarly, what you can do, that you have two strands and bound them. So this interaction will give you a ladder, but a ladder between two strands. Keep in mind, here I'm saying the ladder in the same strand. So say one strand, you simply fold it back, you get a ladder. And in the other part, you are saying that we have two strands and they are coming close. Is it okay? So if we have these informations, now what someone will think, if I keep increasing this first figure, in the first figure, if I keep increasing this interaction energy, what interaction energy is? I'm saying that you have more and more and more energy between these, forming this ladder, the one strand folding it. What do you think? If I increase this in energy, this will be more stable state, yes or no? So I'm simply giving energy, more and more energy. So then this one strand will be folded. If I give that, okay, no, not this, I want the two strands to be together, then I'll give energy, which is called EP. I just gave this name EP and ES just to distinguish between the energy type. So if it is ES, it is between the same strand. And if it is EP, it is between different strands. So now with this ES and EP, can one think about that, okay, might be these EP and ES are nothing but whatever the hypothesis had been made, that, okay, there could be some charge neutralization, there could be some sort of a BSA, there could be some sort of segregation or the Crowders. So is it possible that by simply tuning this ES and EP, we are modeling, we are somehow capturing the surrounding environment of the cell? So if I can do that, in that case, we can simply see a very simple-minded approach. Here, I really think that those of you who are not from the, who has some problem, please ask, because otherwise it will be simply a talk from my side, and I know my work, so I don't have to convince myself again. So I really think you should ask. So when I say EP, I simply means that two strands are close, two strands. When I say ES, I say that each strands get separately close. My strand's name is A and B. So when I'm writing NAB, it is simply meaning two strands coming closer. And when I'm writing AA and BB, it means the same strand can fold over each other. So if I can look at this expression by putting these energies, what is the simplest thing one can think about? That if the EP is 0, let us say EP is 0. EP is what? Bringing two strands closer. Okay. So if I'm making EP 0, what I'm doing? Anyone? What I'm doing?
I'm basically promoting the interchain folding.
I am promoting the interchain folding. So not promoting means, in what sense you are saying promoting interchain? Interchain means
like, yeah, I mean the same chain folding with
exactly.
Like,
so intrachain. So in my words,
sorry,
it's intra.
intrachain.
Okay. So the whole important point is that if I'm keeping EP 0, then I'm saying, no, you have two strands, you can't come together. You are only allowed to fold on itself. That is the role of the ES. Now, if I have ES 0, let us say that intrachain energy is 0. So then I'm simply saying what? That you can ES 0 and EP very large. Then I'm saying, okay, these two strands, you simply come and bound. Okay. And if we have some sort of extremes in these manners, which is just EP and ES, and what will happen if we have both the energies 0? Any guess? Any guess? So if you have both the energies 0, I'm allowing nothing. It's just a simply floating. Two strands are simply floating without forming a structure. Okay. And these 4 states, I think just plug in the values and give a simple physical thought. You will get this some sort of intuition at what type of state you are thinking about. Okay. And if both are the infinity, like both are numbers are very large, then I'm allowing this also, and then allowing the folding on the intrachannels. So by varying this EP, I'm simply saying that I'm trying to mimic the environment of the cell, which is definitely difficult, not easy one, but a simplest way to model the environment. That by some means, how exactly this EP and ES is changing, that I'm not saying. But of course, I know that by changing the surroundings, somehow one can control these parameters. And if you look for this phase space in the physics is like, all the conformations, that which type of conformation this system will have. This is called phase space. So how you'll get the phase space is a simple one. So this is called the free energy balance. So for example, if the two states exist together, so I simply name them 1, 2, 3, 4. One is this ES and EP0. ES and EP infinity is this second state. ES infinity and EP0 third state, and fourth and fifth will come. But important point is that at these boundary lines, this is what we call phase transition. Okay, so phase transition is going from one phase to other phase. And that happens in the biology. So you can simply, like all the talks we have listened, that you can't have just one state. You can simply go from one state to other state. So if you have the free energy for a state 1 and free energy of the state 2 or 3, so if you want to see the transition line, where the transition line is, then you can simply compare these two free state energy. That's all you have to do. And I assume that, okay, let us say the entropy of the state 1 is 1 and entropy of the state 2 is 2. And this is nothing but F is H minus Ts, this enthalpy entropy competition, free energy. So if I take the state 2, where two strands are simply bounded, no intra-chain folding. In that case, I'm saying that, okay, you have the entropy of the state 2 and you have only EP. So how many bonds they will form, depending on the number of beats involved, you will have an EP contribution, and this is the free energy of the state. That is state 2, and similarly free energy of the state 1, because there is no interaction, nothing in the terms of energy, they are just free, neither intra-chain nor interchain, it will just have entropy. And at the boundary, what will happen, you will get some condition from comparing the F1 and F2, you will get the EP, which is a constant. Why constant? Because entropy of the state 1 and 2 will not change, and number of the beats will not change. So this number will be constant. And whenever we have a constant in a phase diagrams, what it means, it just means a line, just a line. So either it can be ES line or it can be EP line. This is just a simple thermodynamical argument. Now, when we did the simulation with this with the interaction potential, we also got, so for example, this is nothing, this phase diagram is from the simulation where we are monitoring the fluctuation in energy. Fluctuation in energy is actually giving you the phase transition, and we got this boundary. So one thing we are very clear about, that this constant line, this lines, the constant line slopes, EP is constant or ES is constant, is good enough because they are coming from the thermodynamics, competition of entropy and energy. And however, with what interestingly simulation gave more than these free energy thing, is there some sort of new things appearing. There are new phases in the phase diagram. So think that we have two energy type of energy, ES and EP, where we knew that when we do the normal entropy, the free energy calculation, we will get these boundaries. In principle, if there is no simulation you must have performed, you must have got just a plus sign in this phase diagram, okay? Because they are constant. But if we did the simulation, we got that there's something extra, which is in terms of the phase 5 and phase 6. So let us see what is this phase. Ashish, you have to please tell me about time also, because I may be slow. So
yes, ma'am, you have
Ashish,
about 15 minutes. Yeah.
Okay, perfect. So let us see this very quickly, that there are these states we already predicted, and the new state which we are saying, that when we talk about the compaction, that there's a new state appearing, and that new state is something like this. This is called sheet. So your DNA means, this is what another analysis we are doing. Hopefully we'll see something more nicer. So your DNA, which are actually just a helical structure, of course it's a helical structure. When you are in this region, some sort of intermediate regions, it really forms a parallel straight, like a plain sheet type of structure. And why this structure is stabilized? Because if you have a again, going back to the entropy, if you have a plain sheet, it can do many folding events, more number of conformations, and conformations are always linked with the entropy. So this new state is appearing because you have this entropy, the entropy coming in the simulation, which were not there when you were doing simple free energy calculations. So this simple-minded approach with varying the EP and ES, you see that, I don't know this movie will play or not. Oh, it's playing, perfect. So you see that we get this by simulation. Either we start from a collapsed compact state, we will always get a intermediate state of this sheet. And this sheet probably will be, yes, here. You see these sheets? And that is really nice about the dynamics part. This you can never get if you are doing the Simple thermodynamical approach where you lose the dynamics. So here also you see the seat. So there is an intermediate state appearing just in this EPES phase diagram, and EPES, I'm telling you that this is the environment, the surrounding environment of the cell, which of course, not I'm saying that exactly because of this reason, but this can be tuned. So in principle, if someone can tune it like this, they will definitely get this type of structure inside the DNA. And of course, this is another thermodynamic analysis where we said that if you know that seed will exist, you can definitely predict this structure. But until unless you know that there is a structure like this, you can't predict the structure. So this is the simulation benefit that you got extra informations. Now, again, question, we have a compact state, folded state, and the unfolded state. Can we get the phase transition? Can we get the melting where we don't see the intermediate state? And the proposal we gave that, of course, if you tune your EP and ES in such a manner where you can only see these two states. So it is possible. So cell might have some ways to do this. I don't know how, but they know that which path they have to propagate so that they can, or the surrounding they have to arrange in such a manner where you can go from one state to other state without encountering. Because if you do any other line here, you know, you always get more state in between. That is not required. For accessing the sequences, you have to be here or here. So the point is that cell might be evolved, or they might have some ways to do this, just a physical intuition for that change the interaction energies inside this, or effective interaction energies, which can control this behavior. Okay, so let us very quickly give me one more example. So probably you all know the circular DNA because this has become very famous these days, is it? So circular DNA is in the mitochondrial DNA and the plasmid DNA, and also I think these days this is some called extra chromosomal DNA for cancerous cell. There are like pathogens, and the whole exercise was that this can one detect the cancer at the early stages if they can guess the circular DNA. There are more number of circular DNA. So this is a very recent talk, and I was very happy to see this and try to connect with this computational approach or the physical approach, that why circular DNA, why the extra chromosomal DNA is more abundant in the cancerous cell. Because for example, if we are humans, our DNA is linear, perfectly linear, but except in the mitochondrial, all the cancerous cell will have this extra chromosomal DNA. What is the reason behind this? Why? So would anyone of you like to tell me the reason? Of course, it is there, that I agree. Why someone will think that this will be there? What is the first things? Anyone? No?
Ma'am, maybe because like in case of humans I'm talking about, there are the like tails. We have the tails in the like what the tails are called, like due to topoism, I forgot the term. Telomere, yeah. So like we have the telomere tails. So in both the ends, so maybe because they are like complementary, they can form the ring. So now the question arises, like maybe like thermodynamically this complementary formation is being like selectively like promoted so that they
Okay.
circularize.
Perfect. Vishal, may I ask a question? So you have a trained biologist or like something? Just a quick.
I'm like I have
Or
my
bio.
bachelor's and pursuing my master's in biochemistry.
Perfect, Vishal. So at least I think this is what I try to understand. So when you see the problem and when we see the problem, so we have to talk more because we have seen the problem in different ways. So when I see this as a cancerous pathogens, I really see that why in the cancerous cell there is more circular DNA. Of course, this might be that telomeric. So for example, if you have the linear DNA, they get degraded, no? So there's an emphasis on that. This, if their telomeric part is getting degraded, so you will lose the whole information, something like that. So you have to protect it. Now, important point is, so why this circular DNA is problematic? Because it is problematic. That's why it's in cancer, no? So cancerous cell has more number of circular DNA, and the could be one possible reason could be you can't change it, no? It remains more and more stable. If you look at my point of view, I may think that even the mitochondrial DNA, you all must know that mitochondrial DNA is really needed for this maternal inheritance, something like keep going from mother to daughter and all the way. So still, I think it is possible to guess that my grandmother, maternal grandmother and her, is it okay? The argument is fine or not? But
Yes, ma'am.
why? But the important point is why only the mitochondrial DNA? Why not linear DNA? So if
Ma'am,
you
so, ma'am, I guess in case of cancer cells, like the most crucial property is like those cells are like divide uncountably, and
yes, divide
for
uncountably.
that the DNA needs to be replicated. And in
Hmm.
case of linear DNA, the issue is with the end parts. Like the more the linear DNA divides, the end becomes shorter. So in case of circular DNA, they have the privilege they do because they don't have any ends. So like there is no chance that the ends are getting shorter or the DNA is getting
Perfect.
shorter.
Yeah, this is the same hypothesis I also thought, but not in terms of biology. I thought maybe it is really hard for circular DNA to divide or something, like it may replicate more, but it's not like that because there are no tails at all. So they will not degrade. So they will be like sitting stably all the time. It's a ring. No, it's a ring. You keep going in ring.
Yeah, ma'am, it's
So
a ring, but like mitochondrial DNA have a special mechanism of replication, the called as like D loop. So
loop three.
I guess
Okay. So here I think we should talk more. So it will be more clearer. But this is really nice. I think when I see this system, I really think so. Then we simply go back and look for these experiments. That what these experiments are saying about the circular DNA.
Ma'am,
And one thing very nice. Yes.
I had a question.
Ashish. Yes.
Uh, regarding role of circular DNA in cancer, right? So
Hmm. Hmm.
typically when the telomeres become short, right, or
Hmm.
damaged,
Hmm.
then the chromosome ends can fuse, potentially
Hmm.
forming the circular DNA, and that
Hmm.
is associated with cancer. Is that the cancer association that you were pointing out?
Okay, Ashish, probably you know it better than me, of course, but just to indulge in that point, my point is very simple. That why? So when we see the cancerous cell, I think this is the recent study. They said that you can use this circular DNA mapping as early stage of cancer. So this is very famous one that you check it, that if there is supposed to be like cancer development, at least chance of that, you can consider checking of the circular DNA. And there are a whole bunch of experiment. I think in your field also you must be at least knowing better than me, that they are really trying to track that
So
if they can track more.
I'll just share my understanding. So what happens
Hmm,
is cancer, right?
hmm,
Cancer cells upregulate telomerase to
hmm,
maintain telomere length and achieve replication, proliferation kind of things, right? So
hmm,
like cancer cells, the fundamental thing is they are proliferative. They are doing uncontrolled cell division, right? So to
hmm,
achieve that, so when normally in normal cells, when cells divide, the telomere shortens, right? With
hmm.
after
But
uh,
there is no tail. But here there is no tail.
so if cancer cells want to replicate uncontrollably, they have to
Perfect.
upregulate telomerase so that they can proliferate uncontrollably.
Exactly. So that's the point. So if there are more number of circular DNA, exactly point. So there is no tail, Ashish. There is no tail. So there is no problem of this degradation. So because there are rings. So we can talk more about it, but let me tell you the experimental observation very nicely. But that is not from biology, you know. That's why I say it's important to tell you that this is just the difference. This is how I see the difference. Now let us look at the PCR. So when the PCR happen, that you simply keep increasing the temperature to get this amplification of DNA. And the same thing happens for circular DNA. You can keep the temperature increasing, and you can get many copies. Now the important part is sitting here. So there was an experiment long back, and the experiment was very simple. So they have a T7 phase DNA, which is a linear DNA, and the intact polymer and nicked polymer. So this is just intact polymer is nothing but a circular DNA which is closed at the end. So they have some linking enzyme, and then they have nicked polymer. Nicked polymer is nothing but they cut one of the bonds. They have topoisomerase, and then they just cut one of the covalent bond. And this behavior you see, it is nothing but temperature and the percentage of base pair opens. Okay. So what is one interesting thing that when you have T7 phase, this is T minus TC. So if you simply do the T version, it is they are mapping. So you will see that this blue one is shifted higher. So it requires higher melting temperature. to melt the circular DNA, you need higher temperature than the linear DNA. And that click observation is really nice because I can definitely think about the higher melting temperature, because as soon as you close the end, the entropy gets reduced. It has less entropy than the linear DNA. And if the entropy is less, then if you talk to anyone, they will say, okay, fine. If the entropy is less, in that case, you require lower melting temperature, higher melting temperature in comparison to linear DNA. So my point here was that, is it like that circular DNA, because there is no telomeric tail, they are simply bound, there's no problem in telomeric degradation, is it possible that they're giving you more stability, longer stability than the linear DNA? And the experiment supports. We still need the biologist consent that, is it okay? So more stability because of the circular DNA, and that's why they are keep, otherwise they could have degraded. No, if they could have simple linear DNA, they could have simply cut in short. But with the circular DNA, you still have this option. Okay, so how much time we have, Ashish? Accordingly, I can simply tell you the results, or am I done? Oh, it's 1.
Yeah.
Oh, okay. So let me just quickly tell you the story, cut my story short. So, okay. So there are many things, but interestingly, when we were doing this cutting thing, so we thought, why not we do this exercise where we simply make it relaxed? For example, the polymer where they made a cut, it's simply the supercoiling is relaxed. And if the supercoiling is relaxed, what will be the role on this melting? So how much stability it will get? So we did this exercise for many sizes of the circular DNA, and we observed that if you have a shorter circular DNA, you will see the effect. So keep in mind, there is no supercoiling in the circular DNA, it is relaxed. Assuming the case like a polymer 2, I think, where they use the topoisomerase for cutting it. And we also see that we have higher melting temperature. So higher melting temperature for lower lengths. But when we increase that length, number of base pairs in the DNA, we see that they are almost sharing the same temperature. So one emphasis, of course, I know the result, why I have the similar one, but my emphasis is somewhere here for biologists at least, and I will just like to discuss more with them, that it actually shows more stability, much more stability than the linear DNA. And that might be the possible reason that why it is getting abundant, why it is not simply dying out. Means there must be some mechanism to control it. Cell knows that, okay, this is extra, I don't want it. So why can't it be protected? The protection is because it needs much more higher temperature to melt, and that is not available. So somehow this reading is still not clear that how to control this excess. If cell can control the excess circular DNA, probably that can help. But they will not, because problem is that this require really high temperature than the normal linear one to control. And of course, there are many other things. So these are the papers. They are online now, and if someone of you interested, you can go through it. So there are other things. Okay, so let us wrap, and let us take some questions if there are. I'll be happy to answer some questions. And of course, at the end, I wanted to show, which I couldn't show, that when you do the modeling, this is not just a modeling which is going nowhere. Probably the best part of the modeling, that when you develop a model, like I said in the very first slide, and then that model is collaborating with the experiment. And there are models which we developed, and recently we collaborated with the experimentalist, and so these are the experimental results. So then you have the best part of your model. You predict and the experimentalist verify. So this is a JMB, I think somewhere. Yeah, here. If you want to see, you can see this paper. So we have developed a model for diffusion of the SSB binding proteins. Again, this gives the stability to single-stranded DNA. And it shows a quite similarity with the experiment. And we are happy that model is established. And we can take it further for prediction. It is at the prediction stage. Okay, so thank you. And these are the people who I should acknowledge first, and then I'll take question if there are any. Questions? No?
Hello. I had one
Yes.
question. It just felt a little counterintuitive to me. In the case for circular DNA, if it is more stable, than a linear one. But in order to have cancer, the biologists are saying that we need uncontrollable
No, so I'm not
cell
sure.
division.
Okay, hold on. So this is the important point, because I have to add in here. I'm not sure that they want more from linear or they want more from circular. This is important. This, I think someone should tell us that what we need more. So in uncontrolled replication, for example, suppose we have more and more number of DNAs. So is it for a linear one or the circular one? Circular one might be just a consequences. For example, if there are more number of linear one, and someone can simply come and close the linear into circular, then you can't, then it will useless, no? Then it will require more temperature to melt. How it will melt? So let us say example that we have a linear DNA, which is a normal functional DNA in the human body. Okay. So by some means, for example, it happens to be that it replicates and work. Now, if it replicates and by some reason it folds back, because I'm not sure that the circular DNA is required for functioning in humans. They are extra chromosomal. So I think more replication or more abundant are not the circular one. Probably the linear one, the one which are dysfunctional, they are getting more in number. And probably they are getting in the circular one. So it's again, it's my viewpoint. And because that's the way I see. I'm not saying that circular became linear in the humans. No, they are not. Our functional DNA is still linear DNA.
So,
Is it
ma'am, I
okay?
want to add some more points to your
Sure.
follow-up.
Perfect, sure.
So, like, I have another viewpoint. Like, in our stem cell, like the telomerase is very much active, which eventually become like its expression eventually like decrease as we like grow and the cells differentiate. So maybe there is a chance that in cancerous cells, this telomerase become like more, starts becoming more expressed in the cell, which actually prevent the ends or like ends become shorter, which is like
Oh,
might be a possibility.
okay. I don't know. So this is my maybe more, but thanks for pointing this out.
And
We have many things to check, but I really think that this will be only possible if we have a crosstalk. You know, the crosstalk that people, at least the students, we are hoping that it's really more talking about their goal. But thank you, Vishal. It's really nice. I'll check it out.
there's
Anything else?
I would just like to add in cancer, right? I also don't know a whole lot about the exact
Yeah, so we should talk, Ashish.
no, I just wanted to add one thing. Let me complete. So there is something called ECC DNA, extra
Extra
chromosome
chromosomal, yes.
circular DNA. So
Mm.
that also plays a role. So I have been reading about it in a couple of papers and stuff, but haven't delved deep into it. But
So,
ECC
no,
DNA, so the
yes.
way telomeres are involved or implicated in cancer is also that ECC DNA can replicate independently of chromosomes. And these
Oh,
circular DNA structures contain oncogenes. You know, there are certain genes which kind of mutations in those are responsible for
oh.
considered cancer onco drivers or cancer drivers, right?
So the
So,
circular DNA have them.
right,
Oh, nice. Nice to know it.
right. So it can, I mean, it's known to contribute to progression and also like drug resistance or chemo resistance.
Ah. No, that resistance I can see anyway, because the way I see that they have more stability, means at least the experiment and the simulations. So they have more stability. So the resistance will be there. So you can't simply do anything like a normal linear DNA. That's for sure I know with this setup, but established setup. But I think we need more input to go further. That what we are. What we further can add to the biology. So these are all simply like the questions motivated with biology. Okay. And I really think that there are something which could be add on. Like yesterday, these questions are important because there are many things which we can add on. But specifically to biology, can we address everything? That is a long way to go. I don't think until unless there are cross talks, it will be difficult. Okay. Anything else? No, probably. Ashish,
Final call.
is it possible, Ashish, that at least at the end you show me the faces of the students?
Ma'am, it's not up to me. Can everybody switch on their cameras?
At least I see that who they are. No, probably I can't see anything. I don't know.
Just for, I think it'll be good to,
Yeah, because I at least do this.
I think video is earnable from the host side.
okay,
Oh, okay, fine.
sorry. No, it should be, okay. Can you switch on now? Sorry.
Vidisha, I can see.
Yes,
Vidisha,
yes.
whom else I can see? They
Dr.
are
Shraddha,
really.
Gaurav, Richa, Akhil, can you switch on?
Vishal, I can
Thanks.
see. Okay. Okay. So I think anyway, we shouldn't pressurize. Let it be liberal. Thank you so much, all of you. Okay, Ashish, we are done.
Thank you for a great talk, ma'am. And I can't
Yeah,
say
we will.
look forward to hosting you because you are at Ashoka. So,
No, we will meet. We will
yeah,
soon meet. I think it was a busy time, but hopefully I'll see you sometime.
okay.
Okay.
Great.
Thank you,
Thank you.
Ashish.
Thank you.
Thank you.
Uh, Attendees, we'll reconvene at 2 o'clock. Please join around 1:55. Sorry about the video. I had locked down the Zoom to prevent any misuse. I forgot about that. So uh, see you at, so we have
Okay.
2 final talks. Thank you, ma'am. Yeah.
Bye.
We have 2 final talks. The first talk after lunch is by Professor Chirag Jain, who is actually, if any of you have used software for genomic analysis, especially sequencing analysis, Professor Chirag actually may have written or be a co-author of one of those packages, or his group at NHGRI in the US. So he was uh, perhaps the best research on sequence software tools for sequence analysis and algorithmic techniques used in those tools. So that should be exciting. And finally, but certainly not the least, we have Professor Durba Sengupta from NCL National Chemical Lab, Pune. She'll be our final speaker. So we look forward to having you join us at 2 p.m. See you then. All right, Professor Chirag Jain. This is Ashish from Ashoka. Can you hear me?
Yeah.
Right.
Yeah, hi Ashish.
Hi. Just an anecdote. You may not remember, like, I think during the pandemic, you were teaching a course and I had emailed you. That time I was not with Ashoka, but I was interested in genomics. So I had emailed you, like, if you could put it up on NPTEL or something.
Okay, interesting. So actually, coincidentally, we are starting a NPTEL course on yeah. So we'll have NPTEL course ready by January. So it will
Okay.
start in January. So just yeah, thanks for bringing that up. But good to
During pandemic, there was a phase, right, where in-person classes were not happening. It was on Zoom, right? So
correct.
I had requested if you could extend it to people outside campus, but I think there were some technical issues. But yeah, I'm really excited. Sorry, you were saying something.
Yes, I think because back then I was also starting the course and I was not confident enough. That was probably the primary reason. But now I think we have done the course 3, 4 times. So then we thought we can do it on probably on a broader platform. But thank you for encouraging me in
No,
that
sorry.
sense.
I think if I remember right at that time, like your PhD involved working with NHGRI, right?
Yes.
Right. So, I mean, I think you are just being humble when you say you are not confident enough. If you have been at NHGRI, then I think, you are perhaps at the frontier and the state of the art in the field. So, but really excited to have you here
Thank
today.
you. Yeah, thanks. Shall I check my screen sharing? Is that okay? Just to uh,
Yeah.
yeah. Yeah. So let me just make sure that things are working okay. So okay, this is only sharing. Let me stop share entire screen. Yeah. Okay. So now, so for example, if I go to the full screen and then I
Correct.
move my slides,
They're
are you able
moving.
to see?
Yeah.
Okay. And you
Correct.
can see my pointer as well, right?
Can you maybe change it to red, perhaps, just so that it's better visible?
Yeah, though that setting,
Just right click. I think there's some pointer. If you go to, uh, is this PowerPoint or Keynote?
this is Keynote. This is Apple Keynote.
Yeah.
Uh, let me, hmm,
In preferences, I'll just Google also. Are you okay changing? Okay, you figured it out. Okay.
yeah. So this is better.
Yeah, this is much better. Yeah.
Okay,
Yeah. So we'll give it maybe 3, 4 minutes and then start.
sure.
But I think, yeah, it's okay.
I'm curious if all the attendees are attending online or some of them are attending from in person at Ashoka.
Sir, I think it's mostly online
Okay.
because also it's summer at Ashoka, so like faculty is also either traveling and students are off to internships and stuff.
Okay, got it.
Are you on campus during the summer? Are you also traveling?
No, yeah, I was traveling for a brief time, but yeah, mostly on campus.
And just I had written in my email, but just double checking, you are okay it being recorded and live streamed, right?
Yes, yes, yeah.
Okay. Because we want some like the PI of the center, Professor Gautam Menon, and some other faculty may not be able to join live, but they would love to watch it later. And
Sure, sure.
so maybe we'll start in another minute. Okay, I'll just give it 2 more minutes for people to come back from lunch, and then we'll start.
Okay.
So, welcome back, everyone. We are now going to proceed with the post-lunch sessions of day 2 of the workshop. And we are very excited to welcome Professor Chirag Jain from ISC Bangalore. So I'll quickly read out his introduction. You were there during the break. Some of you may have heard that he did his PhD at NIH and at GRI. So if you're interested in bioinformatics and genomic software tools and algorithms, you shouldn't miss this talk. So I'll quickly read out his bio. Professor Chirag Jain is an assistant professor and India Alliance Intermediate Fellow in the Department of CDS, Computational and Data Sciences at IISc. He leads the ATCG group, which develops algorithms, theory, and software tools for data-intensive problems in genomics. Prior to his appointment at IISc, he worked as a postdoc fellow at NIH. He completed his PhD in 2019 at Georgia Tech. So very excited to have you, Professor Jain, and please, just a quick note, please let us know when, are you okay with questions throughout the talk? Or?
Yes, I definitely encourage everyone to ask questions during the talk. So you can just please interrupt me. So I'm not able to see any raised hands, but you can just speak up and interrupt when you have any questions.
Right. I'll redirect them to you and interrupt you if required.
All right. Okay. Thanks. Okay. So thank you and thank you for this opportunity. Actually, I'm excited to present some of our work at this workshop. And so today's topic will be algorithmic challenges and solutions for the genome assembly problem. Now, I just want to say a little bit about my group. So I've been at IISc since 2020, and we are the group here known as ATCG Lab. So it stands for Algorithmic Techniques for Computational Genomics. My primary background is in computer science. So I'm really interested towards designing algorithms for problems in life sciences. For example, currently we are focusing on building bioinformatics algorithms, theory and software for both fundamental problems as well as problems of clinical relevance. So the fundamental problems include building and using pangenome references, how to do de novo genome assembly, or genome resequencing at better speed and better accuracy. On the clinical genomics front, we are looking at cell-free DNA analysis and also analysis for rare disease diagnosis. My today's talk will focus on the genome assembly topic. And for this presentation, I will walk you through the overview of this problem, maybe giving you some history, what this problem means, what has been previous work, what are the open challenges that are still unsolved and that require you to think more, and maybe some of you will solve them. I'll also talk about, once I've given the background, I'll also talk about one recent research work that we have done on this topic. This is unpublished work, but this is currently under revision at a journal. And finally, if the time permits, I'll give a short demo of the software that came out of this research. So to this audience, I don't need to tell that the human genome comprises 46 sequences. So basically, we say that we have 23 pair of chromosomes. And for each chromosome, we inherit 2 near identical copies, one from father and one from mother. If I take a look at the total sum length of all 46 sequences, that is about 6 billion characters. Now, when we do whole genome sequencing, we can take a blood sample of a patient, we run it through a sequencing machine. Ideally, what we would like is 46 sequences, one per chromosome in a file, in a text file, right? That would be the ideal output, that to know what the genome of the patient is. But in reality, what we get is small pieces of DNA. So depending on the technology, the length of these pieces can vary. So at the time of sequencing, we get these short pieces and we don't know where these pieces belong to in the original genome. And we also don't know whether the piece comes from the paternal copy of the genome or the maternal copy of the genome. So the problem of genome assembly is to reconstruct the original genome from these short fragments that we call as reads. Now, I have to emphasize that the genome assembly problem is not a new problem. In fact, this is a very old problem, and I'll give you some historical context of what has happened in the past. First, let me describe what are the challenges involved in this genome assembly problem. And for this, let me draw an analogy from genome assembly to this jigsaw puzzle. So you can imagine that when we are solving this jigsaw puzzle, we are given as individual pieces, right? And we have to find the correct place at which to place the puzzle piece. Similarly, in the genome assembly problem, the equivalent thing we have is the set of reads. We want to reconstruct genome instead of a picture like this. And we have to consider pairwise similarity among the sequences in order to figure out the order in which those sequences should be placed. So one can imagine looking at the overlaps among the reads and figuring out, okay, what read should appear next to my current read, for example. So this analogy is frequently used to describe the genome assembly problem. And what we are looking forward is to reconstruct the entire picture in this case. So what are the challenges? So to understand the challenges, again, the jigsaw puzzle helps. So the biggest challenge in the genome assembly problem is that the genomes have too many repeats. So imagine that when you're solving a jigsaw puzzle where there are many identical patterns in the image that we have to reconstruct, then we will face more difficulty in figuring out where the piece should go, right? So similarly, here in the genome, we have too many repeats. We have too many pieces. Basically, we are assembling 5 to 50 million reads, right? There can be missing pieces. For example, there may be some regions of the genome which may not be sequenced, and most pieces are damaged, which means that in practice, we will have sequencing errors in these read sequences. And depending on the sequencing instrument, the error rate can differ. These are the 4 challenges. And one last challenge that I have to remind is that we are solving the de novo genome assembly, which means that we don't have access to the reference genome. And that basically means that imagine you borrow the puzzle game from a friend, but they don't give you the box on which the final solution is displayed. So all we have is the set of reads, and we don't know how the final genome is going to look like. So the problem of genome assembly is to take this set of reads and to reconstruct the entire genome without having a knowledge of what the genome is going to look like. So I want to give at this point a historical perspective, right? First, why is this problem important? Well, this problem is important for answering fundamental questions in biology and disease. You can imagine using this for building high-quality reference genomes and pan-genomes, reconstructing metagenomes, transcriptomes, studying genome evolution, phylogeny, structural variants in a genome, and also to reconstruct genomes of non-model organisms for biodiversity and conservation efforts. So there are many applications, and that's why genome assembly has been a fundamental problem for bioinformatics. And it has been a fundamental problem for the last 50 years. So it turns out that the first genome that was assembled was in 1977 by Fred Sanger. And that was a viral sequence known as 5X174. And even then, in 1970s, people had already been working on building computer algorithms in order to assemble these pieces together. So this problem has been active for 50 years now. And looking at the timeline, in 2003, the research community assembled the Human Genome Project. So the Human Project was completed. In 2014, between 2003 and 2014, the cost of sequencing dramatically dropped. So large number of genomes could be sequenced. And I should say around this decade, also we saw significant improvements to sequencing technology in terms of the read lengths. And that also helped enable the first complete and gapless human assembly in 2021. So due to repeats and due to evolving sequencing technology, It has been a long historical context that this problem has. I'm happy to pause and see if there are any questions.
So the one that happened in 2021, that was called the T2T consortium, right?
Right. So this is by the telomere to telomere consortium. Yes. Yeah. So that's why the name comes from. So that's why the these genomes were referred to as telomere to telomere, because telomere is one end of the chromosome and other end of the chromosome is telomere. So the genome reconstruction is happening from end to end without any gap. So this was the first time in 2021 when the first human genome could be assembled end to end with various advancements.
And there were, would it be fair to say there was a mixture of sequencing technology advancements and algorithmic genome assembly advancements?
Yes, absolutely.
Bhavesh, if you want to ask your question, sir, one of the attendees had a question about Bhavesh.
Oh, yeah. Thank you. Thanks, Ashish. So I had a question about the need of doing all this assembly. Seeing that the reads are fragmented. So I posted in the chat and I got a reply saying it's because of the way the sequencing is done. But in 2021, this gapless assembly. So I'm not from a biology background. I'm a computer science PhD student here at Ashoka. I'm just trying to understand this. So why these pieces are fragmented instead of, can we not do the whole thing sequence, or is it expensive, or is it not technologically feasible?
Yes,
Yeah.
currently it is not technologically feasible. So the process of DNA sequencing, library preparation, that itself involves that in that process itself the DNA gets fragmented, and so yeah. So due to technological limitations, but maybe in future it may be possible to read entire genome. So that's I don't think no one has proven that it's impossible. So it may be possible, but right now due to technological limitations, it's not possible, and we still see whenever we sequence the genome, we get short pieces out of it.
Okay, thanks.
Okay, all right. So I'll move ahead. So this was the achievements that happened in sort of the assembly space, but I also want to give the algorithmic history. So that, like, again, the algorithmic history is also very old, and there is a lot of context. So initially in the computer science literature, the initial attempts to solve the genome assembly problem were using string-based problem formulations, where some of the original formulations that were given to solve this problem were basically based on finding shortest common superstring, or using greedy algorithms, maybe basically looking at the highest suffix-prefix overlaps among the reads, and maybe pairwise joining them until we end up with a single string. The other side that also got popular at the time was graph-based algorithms. And in the graph-based algorithms, you can imagine building a graph structures where the individual nodes represent some strings, and the directed edges represent overlap relationships among the strings. Right? So when I say overlap, I mean suffix-prefix overlaps among the strings. So there were two kinds of graph-based methods proposed, and these were known as string graph, and the second was known as De Bruijn graph-based approach. And actually, both ideas were presented at a conference in 1994. And I should emphasize that since even today, all the practical assemblers that are being built on top of these two graph-based methods. So on top of these graph-based data structures, several heuristics have been introduced in the literature. And you can see that many of these assemblers are either using De Bruijn graph-based approach or a string graph-based approach. And yeah, and in combination with many useful heuristics to deal with real data. So some of these are genome assemblers, and some of these are, like, for example, Trinity was a transcriptome assembler. So, okay. So given this context, I also want to emphasize that the sequencing technology has been evolving. So, and the why this is important, because if you so post 2010 we saw the uh, long read sequencing hitting the market. And why? What do I mean by long read sequencing? Basically, so there's we can think of two types of high throughput sequencing technologies. One is short read sequencing, where the fragments that we get out of the genome sequencing experiment are of fixed length and they are very small. So they're like, for example, a typical length is 150 base pair. So reconstructing 6 billion base pairs from just pieces of 150 characters is extremely difficult problem. What helped genome assembly, the field of genome assembly, is the advent of long read sequencing, where longer pieces were now available and average length of these reads was more than 10,000 characters. And why this matters? Because now you can imagine that the puzzle that we are solving, now we have access to longer pieces, and it helps because now using longer pieces to solve the jigsaw puzzle is relatively easier.
And I had another quick comment and question. Sorry to interrupt you. I was just wondering, there's this piece of software by Google Research called Deep Variant. So I just want, will you talk about towards the end about the impact of deep learning and machine learning on genome assembly and variant analysis and stuff?
Sure, I'm happy to discuss towards the end. But the deep variant will not come into the picture here, because deep variant is based on deep variant would be used when you rely on a reference genome. So suppose when you are doing genome resequencing and we want to use the human genome reference, so there we would align the reads to the human genome reference and look at the alignments and use deep learning to assess what changes are sequencing errors and what changes in the reads are actual mutations. So there, yes, deep variant is important, whereas the talk today will focus on the de novo assembly, where we assume that the reference is not known to us.
But practically, when in biology we talk about sequencing, we almost always align it to the human reference, right? GRCH38 or...
Yes. So for human genome sequencing, that is true. That majority of analysis being done uses the human genome reference. But as I mentioned, that suppose you want to study a region which is absent in the human genome reference, because the human genome reference currently is not complete. There are many unresolved regions in the reference, and there are many regions which are difficult to align. So even in human genetics, having a de novo assembly is useful to study large structural variation and rearrangements, which would not be possible using a resequencing approach.
And which are very relevant if you are studying disease or cancer, these rearrangements and SVs or structural variants. Okay.
Correct.
Thank you.
But yes, and outside human genetics, one can think of assembling or reconstructing new genomes where a good reference is missing, right? So we have so many species that have been for which a reference genome is not available. So a quest is also to have a high quality genome knowledge of all the species on Earth, for example.
So genome
that would require
is to make the first reference genome for a species for which it doesn't exist.
Correct. And also, if you want multiple references for a species, for example, if you think that, okay, within the human genome community, we want one reference from coming out of Indian, or for each population, we need a high quality reference. That could also be a way forward. So that we call as pan-genome reference, where you want to make sure that we have the diversity captured when we are talking about a reference.
Very interesting. Thank you.
Okay, thanks. Yeah, thanks for the question. That was very relevant. So moving ahead, so you can see, so I was talking about the long read sequencing, where we can fragment longer sequences now, and you can already see the impact that the long read sequencing had. So this is on the left is the human genome assembly in 2001. So this was the human genome assembly that was being produced by the Human Genome Project, and you can see that the gray and black markers show the breaks in the sequences. So the black-gray is sort of telling you that the sequence is broken due to repeats and many other challenges. Whereas on the right, you can see that with long reads, the contiguity of assembly has significantly improved. The white regions represent the regions that are still missing in the assembly. So on both sides, you see the white regions of the chromosomes, but you can see the long stretches of either gray or black on the right-hand side, telling you that the contiguity of assembly significantly improved when long read sequencing came into the picture. Now, this was 2019, and the right-hand side assembly is also by the T2T consortium. What has happened in the last 5 years or so is that the accuracy of long read sequencing has dramatically improved. The initial versions of long read sequencing instruments were infamous for their high error rate, whereas post 2020, the accuracy of long read sequencing is now hitting 99% or even better. So for example, if you look at the two major sequencing technologies, PacBio and Oxford Nanopore, both have provide options to support long read accuracy of 99% or more, whereas previously the accuracy was below 90%. Now, this was what enabled this this was a big factor that led to the first complete assembly of the human genome by the T2T consortium, because they were able to use this high quality long read sequencing to reconstruct the human genome. And this improvement in accuracy actually spurred development of new long read assembly methods. Why? Because of two reasons. One is phasing. So previously, the state of the art in the assembly was to produce 3 billion characters as output. Basically, the assemblers would collapse the paternal and maternal haplotypes into one sequence. And what they would give out is a consensus of the two sequences. Whereas now the state of the art has shifted, and the expectation is that the new assemblers will give us the two haplotypes, the two paternal and maternal haplotypes separately. So the output size of the new assembly methods is now 6 billion characters instead of 3 billion characters. The second change that has happened is with the new sequencing technology and assembly solutions, the long and inexact repeats that are significantly longer than the read length can now be resolved, because these reads are so accurate. One can distinguish between inexact repeat copies and figure out how to solve the puzzle properly in the presence of repeats as well. So for those who want to know more about these aspects, I also recommend you to read a very recent review article on this topic by Henley and Richard Durbin that just got published 1 or 2 months back. Now, one can ask, okay, is the problem solved? Is there anything else to do that requires attention, or we just should go home and relax? Turns out there are many things to do. First is that the automated long read assemblers still leave many gaps in the assembly, and there are base level errors made in the assembly. So they don't provide a perfect solution yet. Still, obtaining a perfect deployed genome assembly requires months of manual curation. So one has to use a mixture of technologies, use IGB visualization, and see where the errors might be happening to fix the last errors that the assemblers are making. Third challenge is that these assemblers are driven by many heuristics, and there's a lack of theoretical guarantees. In computer science, we always desire a elegant problem formulation and exact algorithms with some guarantees, but that's missing in the genome assembly space. Fourth is that for genome assembly, especially for de novo genome assembly, the amount of data or the number of reads that we need is significant. So for example, if I'm going to use nanopore sequencing for a single T2T human genome, that would currently cost me $4,000 US to generate the data needed for T2T genome reconstruction. And that's not good for, especially for developing countries where we would want to deploy this technology for on a routine diagnosis basis. Finally, assembling cancer genomes or polyploid genomes is further challenging, because so far we are talking about diploid where you have 2 copies, but as soon as you move to cancer or polyploid, there will be multiple haplotypes that one would want to assemble. So then one has to resolve multiple copies of each chromosome in order to guarantee a perfect solution. So given these challenges, our lab's interest is to build foundational algorithms and theory to achieve low-cost, routine, and fully automated telomere-to-telomere genome assembly of human genomes. So at this point, given that I've described you the open challenges that need attention and what our interests are, I'll move to the second part of my talk, where I will be describing the recent research that we have done on this topic. And I'm happy to stop for a second and see if there are any further questions about what the problem is and any understanding of the problem that you would want.
Had a quick comment / question. There is also, I'm forgetting the name, but there was this newer long read technology, or I'm forgetting if it was long read or short read, which is a big competitor to Illumina, and like they're claiming that they can do it at $100 per genome. Like I was just wondering. Maybe I'll ask it towards the end. I'll let you.
Right.
Proceed.
I think it's molecular, but I may be wrong because we are also not using that technology right now. But
Okay.
yes, you're right that there is a competing technology which gives much higher accuracy of reads or short reads, not long reads. So that is for short reads. Yes.
In long reads, the 2 dominant players are Oxford Nanopore and PacBio.
Yes.
Thank you.
Yeah. Okay. So moving ahead, so before I describe the research, I need to give a little background on what kind of graph methods are used in genome assembly space. So intuitively, you can think of the most intuitive thing that should come to my mind is building an overlap graph where we have sampled the reads from the genome. And now what I would like to do is represent each read as a vertex in the graph. And I would want to show the suffix-prefix overlaps among these reads as a directed edge. For example, R1 is sampled from a region like this, R2 is sampled from an adjacent region. So therefore, I would have a suffix-prefix overlap between R1 and R2, and I can draw a directed edge from R1 to R2, right? So I can build this entire graph. And maybe once the graph is built, I can think of what additional steps are needed to get the genome out of such a graph, right? So given this graph, that's not, so we can build this graph, that's not a problem. But turns out that the graph is very messy. For example, what I'm showing you in this picture is a graph, overlap graph, built from a small number of simulated long reads from human chromosome 20. And what you're seeing is that each sort of colored rectangle is representing a read here, and the black line segments are indicating the edges. And you can see that what you would want is a linear chain-like structure, but what we are getting is a very complex graph topology that is not useful and easy to interpret of how to get the genome out of such a messy graph. So the next step that people would use here is to simplify this graph and maybe try to remove redundant portions of this graph so that it becomes easier to handle. So at this stage, what happens is that in the overlap graph-based assembly algorithms, the next step would be to remove contained reads. So by contained reads, I mean short reads, which are shorter reads which are part of longer reads as a substring. So for example, you can think here that R3 is a substring of R4. So maybe I don't need R3, that R3 is redundant information, right? So R3 is a substring of a bigger read. So maybe I can remove R3 and make my graph simpler, right? So you can see that ATC, AC, this is a substring of R4. So it's already present here. So as a result, I drop this read and I make my graph simpler, right? So what happens as a removal of contained reads is my graph is becoming simpler at every stage. As a next step, I'll also remove transitive edges. So by transitive edges, I mean that these are redundant edges which are spelling the same sequence as spelled by other parts of the graph as well. So for example, when I'm spelling the genome, I can either go from R1 to R4. So I can spell the R1 and then I can move to R4 and then I can continue to spell the genome. Or I can go from R1 to R2 to R4. Either way, I will spell the same sequence. So on this graph, you can see I can go from R1 to R4, or I can go from R1 to R2 to R4. Both ways, I'll be spelling the same sequence as you. So basically the way to spell the sequence is you spell the first read, and then you ignore the overlap, and then you spell the remaining portion of the second read. So in this case, some of these edges are redundant, and I can remove them. And this is a safe heuristic that I'm not losing any information here. So once I do this, I get a much simpler graph to analyze. And if I get a chain-like graph, then predicting what the genome is much easier. So you can see the impact here. So on this small similarity data, once I remove the contained reads, I get a much simpler graph. And if I remove the transitive edges, I almost get I get a chain-like topology, and predicting the genome from this chain-like topology is very trivial because I can go from one read, then I can visit the next vertex, then I can visit the next vertex, and I can continue to spell the DNA characters along the way. Now, what is the challenge here? Where is the research needed? Turns out that in the contained read removal, this step is erroneous. So when we are removing these contained reads, which are part of longer reads, it sometimes creates issue in the graph. So as an example, let me show you this read R6, which is contained in read R2. So here I'm showing you both copies of the chromosome, maternal and paternal. They are differing by a heterozygous SNP, and the rest bases are same because maternal and paternal copies will be near identical to each other. As a result, the read R6 is contained in read R2 because it will be a substring of read R2. Now, in this case, if I remove read R6, turns out I can spell both maternal as well as paternal haplotypes in my graph that I will build. However, in another example, if I'm unlucky and if read R3 is, let's say, shorter, then what will happen is that read R6 will be contained in read R2. R6 will be dropped from the graph. And what ends up happening is that there's no path that spells the paternal sequence anymore. So from R4 to R5, there's no way to go to R7. So there's a gap introduced in the middle, which we call as an assembly gap here. So this is a sort of a major limitation of the overlap graph-based approaches, where even though the method has been known for now 30 years, this has been a fundamental limitation of the overlap graph-based methods that has been not resolved yet. That how do we remove these contained reads properly so that we retain the reads which are actually useful in the assembly and not lose the useful information in this process?
I had a very kind of dumb, naive question.
Yes.
So when we get the reads, do they have information whether it's from the maternal, from the maternal copy or the paternal copy?
No, that information is not available.
Like even in the metadata associated or
No.
anything? Okay.
No, because the DNA sample will have both. And when the sequencing is done.
Is the read a mixture of the two?
So a single DNA molecule will always belong to only either one maternal or paternal copy. So a read, when a sequencing machine reads the DNA, it will read from a single molecule. So a read will only come from either maternal or paternal haplotype, but we don't know which one of the two. Does that make sense?
Right. And are there any, once assembly is done, can we retrace as to which one came from is there?
Yes, that is possible. So for example, if I give you a perfect genome assembly output and I tell you that, okay, this is the maternal copy and this is the paternal copy. And if the read has a heterozygous variation, like it should have some heterozygous information. Otherwise, for example, for R6, I'll never be able to tell.
Can you explain heterozygous variation, like just for everybody?
Yes, so heterozygous variation is when where maternal and paternal copies differ from each other. So for example, here I'm showing you as an example G and T. So at this location, this is known as heterozygous SNP, where both maternal and paternal copies are differing from each other. The other locations are known as homozygous, where both copies are identical to each other.
So post-assembly, one can delineate, but not during assembly.
Yes, one can delineate most of the reads. For example, I cannot delineate R6 even then, because R6 has no heterozygous SNP. But I can delineate R5 because R5 has this T base. So if I do some pattern matching, I can tell, okay, R5 must have come from the paternal copy of the genome. Does that make sense?
Right. No, I had only heard about alignment, and assembly is just like so much more challenging and yeah.
Yes, so it becomes a different problem altogether. Yeah. I see one raised hand. Yeah, please go ahead.
Yeah. So I had a question. Why we cannot delete R6? Sorry if I missed this. Deleting R6 leading into some error. Can you just tell me that?
Yes. So remember that when we are drawing this graph, each vertex is a read, and the edges between the vertices represent overlap information, right? So
Right.
so if R6 is there, then I can represent the overlap between R5 and R6, and subsequently R6 and R7, and the path will be available, right? But if R6 is gone, and R2 is only for supporting maternal haplotype, so R2 cannot be used for paternal.
Okay.
Because
And we don't
R2
know.
has.
And we don't know which one it is coming from, so we cannot be sure, right?
Yes.
Yeah.
Yes.
Okay. Yeah, that makes sense. Thanks. Yeah.
And the other comment could be, can we just retain all the contained reads and then work with the assembly? But the problem with that is this: that if we retain all the contained reads, the working with the graph becomes impossible. So in practice, 99.9% of the contained reads will be actually useless. Only that 0.1% it will create an issue like the one I'm showing here. So this becomes like a needle in the haystack problem, where if retaining all the contained reads is not an option, and finding out what contained reads are useful is also very difficult.
So what do you do then? Like, do you just delete everything and see if the problem occurs, or how is it?
Yes, right. So I'll describe a solution. One other possible solution is that, okay, maybe I can consider rebring all the contained reads back and see if they join two adjacent vertices in the graph. But that's also problematic because sometimes what happens is that there may be two consecutive contained reads that got lost, or there may be three consecutive contained reads that may got lost.
Right.
There are these practical issues so there is an assembler that tried this method of bringing some of the contained reads back, but that has not been found to be useful for working on real instances.
Okay. So do you say it's a non-polynomial time thing then, right?
I think so. Yeah. So if one formulates it, yes, it will end up.
Okay.
Although we made some attempt to prove, but we don't have a proof for proving its interactability yet. But it looks like an intractable problem.
Yeah.
I had another quick question. Can you do some sort of, like, if you had a lot of data of like frame it as a machine learning problem where you have gold standard of assemblies for a lot of previous runs, and that helps you predict which contained reads to discard and which to keep?
That may be possible. It has not been explored so far, and yeah, but that may be possible. It's very, for me, it's very hard to guess whether how much data and what kind of data is needed during the training step to make sure that the model will start.
If you're doing genome assembly for a new species, there may not exist an assembled genome for that species. There might exist assembled genomes for other species, but
yes, but there is work going on building AI methods for other components of the genome assembly problem. And what people there do is that they train on some, let's say, few chromosomes of the human genome, and then they will show the applicability of the model on other species as well. So they try to learn in a way that they can learn the basic properties of the problem and not the species-specific properties as much as possible.
the more generalizable parts of that.
Yes, yes. But there's an active research on that topic. So many problems I can discuss in the end, because there are many sub-problems within the assembly space. And on that, some of them are being looked at from an AI perspective as well.
Thank you.
Yeah. Okay. So moving ahead, I also wanted to show you, the slide I skipped, that in practice, for a deployed genome assembly, this is how the graph will ultimately look like. Like if you collapse the chain-like structures into one node, then what you would get is these bubble-like structures in a graph where one way would spell the maternal genome and the other way would spell the paternal genome. So I'm just showing you a figure from an existing assembler which assembled the deployed genome. And you can see how the graph will look like in practice, just to get a sense of how deployed genome assembly is different from haploid genome assembly. And of course, if you have polyploid genomes, then there will be multiple parallel. One would hope for multiple parallel paths. Okay. So the questions that we were asking in this research problem is that first important question is how frequently do these gaps occur when we are removing these contained reads? Because if they're not occurring at all, then we don't need to worry about this problem at all. Second thing is, if this problem is occurring, how can we make the existing assemblers resilient to this problem, right? So these are the two questions that I will walk you through one by one in the next few minutes. So first, for the first part of the talk, for the first part of this paper, what we did was we identified the goal is to identify the influence of sequencing coverage and your choice of sequencing technology on the gap frequency. For example, the frequency of gaps may differ depending on whether you're using PacBio or Nanopore, or whether you're sequencing at 20x coverage or 40x coverage. So how much data that you're going to generate will also can affect the number of gaps that may be seen in practice. And this problem is actually very challenging because to determine the count the number of gaps, I need to exactly know what are the sampling positions of the reads, what are the read lengths, where are the heterozygous SNPs, and what is the sequencing coverage. And integrating all these parameters together, it becomes a very hard maths problem. So as a first attempt, what we try to do is we simply made some simplifications. So we assume that there's a hypothetical deployed circular genome that has a single chromosome, and it has only a single heterozygous SNP. So you can see there are two copies of maternal and paternal haplotypes, and they only differ at a single heterozygous SNP. We assume there are no repetitive regions, and we assume that error-free reads are sampled uniformly at random from these genomes. So we are making many simplifying assumptions here, but this is to just get a handle on the problem of how frequently would the gaps occur in a genome assembly. And by frequency, I mean that if you think of all possible sequencing outputs, for example, all possible sequencing outputs that one can get in terms of the read sampling positions, out of those, how many sequencing outputs have a coverage gap? It turns out that naively checking all this is not possible because combinatorially, there are exponential number of ways in which I can sequence a genome. The positions of the reads can vary from the start of the genome and the end of the genome. So there are g raised to power n possible outcomes where g is the genome length and n is the number of reads. So we needed a slightly clever solution here. Basically, naive enumeration of one by one is not going to work at all because checking one by one is not going to give us this value. So to do this properly, what we decided is that first we want to define what do we mean by assembly gap, and second we will characterize what exact conditions need to be satisfied for the assembly gap to occur, and then maybe we can think of a clever combinatorial way to compute the frequency of a gap. So to get a handle, let's first define what exactly do we mean by assembly gap due to contained read deletion. So to do that, first what we do is we take a sequencing output and we look at the reads supporting the heterozygous SNP. We look at the rightmost extending read on haplotype 1 that supports the SNP, and we look at the rightmost read extending on haplotype 2 that supports the SNP. So the positions of the rightmost reads are X1 and X2 that support this heterozygous position. So we say that this sequencing output belongs to class X1, X2. This way, we are just grouping the sequencing outputs into subclasses. So this particular sequencing output belongs to class X1, X2 because the rightmost reads extend till X1, and here the rightmost read extend towards X2. Once this is done, for a read sequencing output of type X1, X2, we say that assembly gap occurs if there is if this interval just after the X2, if this interval was previously supported by the reads before contained read deletion, but after contained read deletion we lost support for this particular region. Right? So in the example that I was showing you, you can see that here this is the X2 position, and we lost support for this particular region while we were removing contained reads. So accordingly, we formulated this definition that we would like to define the gap when we lose the support of this region in the genome. Now, once we define this definition, we prove that we give the exact conditions when this assembly gap will occur. So we gave 3 conditions, and we show the if and only if relationship, that these are the sufficient and necessary conditions for the assembly gap to occur. So for example, intuitively, we need the X1 not equal to X2. Basically, these variables have to be different. We need at least one read starting from certain interval until certain interval. Basically, this says that we need at least one contained read. And we don't want any read that bridges the gap.
Professor, we are not able to hear you. Some audio, we can't hear you. I think are you muted, or can you check the audio settings? Still can't, no audio.
Hello, am I audible now?
Yes, yes, yes.
Okay, thanks. Okay. So yeah, so I was deciding that we characterize the exact conditions that are needed for seeing this gap, and subsequently we just give a proof of why these conditions must hold in order to see a coverage gap. For example, if X1 is not equal to X2, but if this condition is violated, then what happens is that we never see a coverage gap, because if this region was supported before, it will continue to be supported even after we remove the contained reads. Similarly, for other conditions, we prove that these conditions are necessary to observe a coverage gap based on the definition that we gave. So once that analysis is done, what we did was we used this, based on these conditions, we defined genetic functions to calculate the number of sequencing outputs which had the coverage gap. And that combinatorial formula gave us the gap frequency that we will see of seeing a coverage gap for a particular technology and for a particular given sequencing gap. Now I'm showing you the plot that we get from that analysis. So what we are seeing here is the gap frequency for 3 different technologies: PacBio Hi-Fi, nanopore duplex, and nanopore simplex. And on the x-axis, you see the sequencing depth on how much data that we are going to generate from the genome. The insights that we get from this plot is that for nanopore, the chances of seeing a coverage gaps are order of magnitude higher than the PacBio Hi-Fi. And for all 3 technologies, the chances of having a coverage gap reduce with increasing sequencing depth. Now, I would like to take a pause and maybe ask you, why do you think one technology would have more chances of seeing a gap versus another? Remember that these technologies differ in terms of the read-line distributions. So the PacBio read-line distribution would look different from nanopore. So what kind of coverage line distribution would make this problem severe? Any guesses?
Can you repeat the question?
So I'm showing you the chances of seeing a coverage gap with 3 different technologies. One is PacBio Hi-Fi, nanopore, nanopore simplex. And these 3 different technologies have different read line distributions. They're all long read sequencing technologies, but they produce reads of different lengths. Okay. So we are using these parameters in the method to guess the frequency of coverage gap, because user has to give us 2 parameters. One is the read line distribution, and the second is the coverage, sequencing coverage.
Right. This is in short read sequencing coverage is usually 30x, right? For
yes, for long read also normally 30x is a recommended coverage in practice for genome assembly.
So the question is, which one of these would you?
My question is, why we are seeing more coverage gaps with one technology than another? So why is nanopore sequencing leading to more frequency of coverage gaps?
Is it like down to the fundamental way how the DNA molecule is broken up into fragments?
So so basically the idea is that it depends on the 3 different technologies are producing reads of different lengths. And the key unique property of the PacBio versus nanopore is that the read length distributions are significantly narrow. So imagine the extreme scenario where all reads are of fixed length. Then we will not see any contained read issue, because all reads are of fixed length. No read will be containing anyone else. So this is the PacBio sequencing is almost close to that extreme scenario where most of the reads are of similar length. Whereas in nanopore, the read lengths the distribution is very wide. So here you can see the x-axis is showing the read lengths and y-axis is showing the fraction of bases. So it's a read length histogram basically. So the number of contained reads or the fraction of contained reads will be much higher in case of nanopore because of the wide read length distribution. Whereas in PacBio, because most of the reads are of very similar length, the chances of a read being contained in any other read will be much less. Does that make sense to everyone or not? So that's why we see that PacBio, the chances of seeing a coverage gap are much less compared to the other technologies.
Does this also imply that so when people are using it for a particular use case, they are more if costs are the same, then I would prefer to use PacBio because my assembler can be simpler.
That actually, for the coverage gap issue, if we just look at the coverage gap issue, then PacBio has an advantage. But for making an overall decision, one would need to look at sequencing cost and what other parameters affect the quality of the assembly. So for the coverage gap issue, like the assembly gap issue that we are talking about, there nanopore reads have a disadvantage. But of course, yeah, the cost and many other factors will come into play when you're deciding between the two technologies. So this is like part of the talk, which was to just show the theoretical characterization of how frequently we will see these gaps. And what I want to, the take-home message here is that the chances of seeing the gaps is much higher when we are using nanopore sequencing because they have wide read-line distributions. So when I move to the second question, where we want to make the long read assemblies resilient to contain the deletion, there we would need to worry about when we are using nanopore sequencing especially. So we need the solution for nanopore datasets more than PacBio datasets here. Because this issue is more likely to occur when we are working with nanopore data. So moving to the second part, the question now is how do we make these long read assemblers resilient to contain deletion? And I'll take some ideas from you as well. So what do you think one can do? Any simple ideas that you can think of what can we do to get rid of this contained read issue problem?
I think Bhavesh said it like, some sort of like, don't like put them in a queue of some sort and bring back some if required and, otherwise discard them permanently.
Yeah, that is actually, that is one option. But what I was saying is that bringing back some is I think one needs to explore more whether one can bring back, how would one bring back, and how would we one check whether a particular containment is necessary or not. But that's a direction that people have already explored, and at least by talking to them, they told us that in many cases it's not working because there can be more than one reads that need to be connected together. And again, it becomes a commentably challenging issue of how many reads do I bring together and how do I judge whether those reads are bridging the gaps or not. So maybe other very simple solution that one can think of is just to break the reads into equal length reads. What do you think? Is that okay? Like, for example, what if I just take the complete read set and I break them into equal length shorter pieces? So that will all get rid of the contained read issue because then there is no read that is contained in a longer length. So, but there is a problem here. And the problem is that we are losing the main advantage of long reads, that the long reads were spanning repeats in the genome. And now we are going back to working with smaller pieces.
But can we, if we add some metadata that all these shorter pieces came from the same longer piece, and we?
Yes, that there may be something possible there. We did think about this, but I'm trying to remember what was the piece that we could not resolve and we had to stop. So yes, that is possible that one can keep the metadata, but the downstream steps of how to use that metadata is again requires some investigation.
And when you say break, you mean like in software, right? Like in the instrument, it's the same read, but the way you process it downstream, you break it down, right?
Correct. It's in the software. So basically, we break the strings and chop it into
smaller equal length substrings. Okay.
Correct. And then pass it to the assembler. So the idea would be to break the reads into smaller pieces and then assemble. But the challenge I want to highlight there is that when we have repeats in the genome, those shorter pieces may not be able to bridge the repeat repeats. So therefore, the assembly quality will suffer if we are working with fragmented reads because we have lost useful information in the process.
Can you opportunistically decide, like, do we know, like, can we detect when it is a repeated region in the genome?
Yes, so that's what I would like to highlight, that that's what we are trying to do. But the only small catch here is that the original genome is not known to us. So we have to do it de novo. And so you almost get the idea. So basically, we have to detect the reads which we think are part of the repeats. And the way to do it de novo, what we do in this work is that we look at the all-to-all overlaps among the reads. And the idea is that if a part of the read is coming from a repetitive region, we would typically see more than expected overlaps with other reads. For example, the green portion here, the read B, you would see more number of overlaps with other reads in the data set. And therefore, whenever we see any region where more number of overlaps are being observed than a certain threshold, we mark that region as a potentially repetitive region. And so the reason of this overlap, so with this overlap information, now we carefully fragment the reads, basically saying that we will still break them into equal length pieces, but we will not introduce a breakpoint whenever we predict a repetitive region in the read. So this is a heuristic basically to solve this problem. But let me know if you have any question. Like, did I describe you well? So what will end up happening is that suppose your original data set had a wide read-length distribution. After the fragmentation, we will make the read-length distribution to a narrow distribution. So you can think of it like making nanopore data similar to like PacBio data intuitively. But at the same time, we are avoiding the fragmentation of those reads which we know are required for repeat resolution in the genome. So that's why you can see that there are still some reads which are kept longer than the default length of 20,000. So there are some reads which are longer than 20, and that's where we must have detected repeats, and we want to avoid their fragmentation. So we implemented this idea, and the way to implement this is that we integrated this idea in existing assembler. What we do is that we let the assembler error correct the reads first. We also let the assembler compute all-to-all overlaps. But once we have done these steps, we introduce our software known as Raft. And what Raft does is it will do the repeat-aware fragmentation. It will fragment the reads, and then once the fragmented reads are available, then we will assemble the reads. So it's a slightly non-intuitive idea because we are saying that previously I said that long reads are helpful, but now I'm telling you that, okay, now I'm going to cut these reads and I'm going to assemble with shorter pieces. But what I want to show you is that this actually, we find that promising results in practice. So the first table is showing you results with similarity data, where we show that the number of assembly gaps when we integrate Raft inside HiFi-SM, which is a popular assembler, the number of coverage gaps come from double-digit numbers to single-digit numbers. So this was a promising result that we got on similarity data, because here we knew the ground truth, where the exact read came from, because where each read came from, because we were doing simulation, and therefore we could count where the assembly gaps are happening. We also tested this method on real data. So we took human genome data from HiFi sequencing, nanopore sequencing, mixture of HiFi nanopore, and 4 datasets which consist of high accuracy, ultra long dataset. And in all cases, what we are reporting is that whenever we have nanopore data, where the coverage assembly gaps are more likely to occur, we are finding that the assembly continuity is typically improving by twofold in practice. And the number of chromosomes that are getting assembled telomere to telomere is also increasing when we have nanopore data. So this result gave us a very positive, so this gave us a positive impact of the algorithm, that the algorithm can be useful in practice. And therefore, when we implemented and released this on GitHub, we also demonstrated that when we were working with real data and nanopore sequencing, we can get much better continuity in terms of the assembly when you are working with graph Hi5SM instead of just Hi5SM assembler. So this is just showing you the ideogram of each chromosome. And again, I'm showing you this individual contigs or individual sequences. The white lines indicate where those sequences are breaking, and you can see the much better improvements in the continuity. We have released this implementation on GitHub, and what we are seeing is positive feedback from others, including Oxford Nanopore themselves, who have used our method and shown good results on human genome data. So what I think in the recent result, what they were able to achieve is 19 telomere to telomere chromosomes reconstructed in an automatic manner using RAF Hi5SM approach. So if there is time, there are two options here. Either depending on the time left, I want to take questions, or I can show you a short demo of the software on a small real data to show you how it is working.
So we can see a short demo and then have questions. We can do both. We have time. We have about 20-25 minutes.
Okay, sounds good. So let me show you demo, and then maybe it will become more clear on this how this heuristic is working. So for that, let me show you. So what I did was we created a small data set from the first 10 million bases of the chromosome 18. So because for the demo purposes, we cannot assemble complete genome because that would take very long. We just took first 10 million bases of the chromosome 18. We sampled. So we took nanopore duplex reads that were sampled from that region, and I'm going to show you the demo from those reads for those subset of reads. Now in this demo, what you can see is I've already done the error correction of the reads, and I've already computed the overlaps among the reads. And I'll feed these two inputs as a input of the RAF algorithm. So let me show you how the RAF algorithm works. So are you able to see my screen? Is the font size visible?
Yes.
Okay.
So the tool has some accompanying visualization also to help you.
Yes, I will show you towards the end. So let me show you the steps. First is that, so let me load the Hi5SM method, and I'll also load the RAF method, and let me also note the coverage of this data set. And first I will launch the RAF method. And what RAF will do is it will take the original sequence of reads, and it is going to chop the reads in a repeat-aware manner. So for this small data set, it is going to, so it is just looking at what threshold it should use to predict repeat region. And that is done. Now the reads are fragmented, and we have the fragmented reads in a file known as RAF fragmented.read.fasta. So these are the reads, individual reads that have been fragmented by RAF. Now, as a next step, I'm going to also see what is the change in the read-length distribution. So for that, I'm going to just run a Python script of just to see the read-length distribution change. And finally, I'm going to run the assembler on these chopped reads. So when I'm running the assembler, I'm passing it the chopped reads, and I'm telling it to use 16 threads, and that's it. So the way assembler would work is that it will compute the overlaps among the reads. It will build the overlap graph, and from the overlap graph, it will produce the longer sequences. So I think it will finish in a few seconds because we're working with a smaller data set here. And ultimately, what I'll show you is I'll show you the graph in a visualization tool. So I download this graph, and there's a visualization tool known as Bandage. that allows you to visualize the graph that we get.
What's it called? The visualization tool? Bandage.
Yes.
Okay.
So I have the assembler has finished. Let me just note down the path where the graph file exists. I'm going to download this file now. Okay, so, and I'm also going to download the plot to just see what is the region change, distribution change. So first I want to show that there is a change in the region distribution. So in this case, because you are working with very small data set, you don't, like, there's not a significant change that you're noticing, but you can see some minor difference. Like, so the black lines show the original read line distribution. This is the read line histogram where x-axis shows the read lines and y-axis shows the throughput. So you can see the black bars here are slightly longer than the blue bars, which means that some of the reads have been chopped and they have been cut into 20,000 length characters. So next I will show how the graph looks like. Because this is a small data set, we don't see much change. But if we were working with a real data set, then we would see significant change in the real line distribution. Let me show you the graph, how it looks like. So for this, I have to just load the graph. So yeah, so this is the graph that you can see. So this graph that we get is from the assembler, and you can see these bubble-like structures in the graph. So let me zoom in. So these are the bubble-like structures that are forming due to heterozygous variants. So one path would spell the maternal haplotype, and the other path would spell the maternal haplotype. And as you go along, you see these bubble-like structures forming. And this is because of the heterozygous variation between the maternal and paternal haplotypes. And as you keep going forward, like again, you see one bubble-like structure, and you keep going forward, and you keep going forward, and the graph ends. So the assembler would basically predict the maternal sequence from this graph and the paternal sequence from this graph through a phasing step. Each of these nodes are going to have some sequence length. For example, the length this purple node I show you, like for example, this purple node, it comprises of 700,000 base pair, which is written here. So this is a sequence that contains 671,588 characters inside it. Similarly, you can see for next vertex, and next vertex. So this is the overlap graph structure that I was telling you about. And each node represents a collapsed version. So this has combined, this is the combination of multiple reads collapsed into a single vertex. So I'll stop the demo here, and I'm happy to take question if there is anything quick question about the demo.
Can we tell from the visualization, whether if some region is a repeated region?
Unfortunately, not in this graph. Let's see. Like for example, I see this region which is not properly resolved. So this must be a repetitive region because you can see that it's still not resolved despite having long sequencing. So if if there are no repeats, you would expect mostly like a chain-like, double chain-like structure. But whenever you see this complex region like this, you would expect that this is happening because of a repetitive region which did not get resolved properly. So you can see that this is not a, like if I want to spell a genome sequence, it would be difficult to tell how to pass through this region. And this is because of a repetitive region in the genome. Does that make sense? Because we are only using first 10 million bases of the chromosome 18, this does not have centromere or some other complex repeats. in this portion of the graph.
Thank you.
Okay, so going back to this demo, so I will conclude my talk here. So basically, what I want to emphasize is that with the improvements in algorithms and technologies, routine generation and analysis of complete personal human genomes may be realized soon. That will also require innovation in assembly algorithms. For this research talk, what I showed you is that the overlap graph-based algorithms are suboptimal when there is significant variability in read lengths. And that's true for Oxford nanopore because the read lengths vary significantly in Oxford nanopore sequencing. And the proposed method that we showed of repeat-aware fragmentation improves the overlap graph-based assembly continuity by twofold. There are two software. One is the RAFT, which I showed you the demo, and the other is for the calculation of frequency of gaps, the combinatorial method that checks what is the frequency of gaps for a particular sequencing technology. Both of the software are available on GitHub, and you are welcome to try them. And the paper describing the research that I talked about today is also available on bioRxiv, and it's currently under revision at Genome Research. I'll close my talk here by acknowledging the students who did this work. So the project was led by my student Sudharma Kamath. He's a PhD student in my lab. He was helped by Mehak, she's a former project staff. And this project is in collaboration with Professor Devnath Pal in CBS. And I will just close by thanking the funding agencies and happy to take more questions. Thank you.
Thank you, Professor Chirag. Please, attendees, feel free to ask questions. I had one, sir, kind of unrelated to your talk, but in the first part of your talk where you had introduced kind of the research interests of the lab, you had mentioned also rare disease genomics.
Yes.
So I was curious, so that's an interest of mine as well as one of the PIs, co-PIs of the center, which is organizing this talk. So I was just wondering, so there is this notion of diagnostic odysseys in rare disease because they don't get diagnosed for a long time because of lack of variant interpretation and clinicians and trained genetic counselors and all those. So I was just wondering, have you done some, like, has some of your research been on that? Could you talk a little bit about that?
Right. So actually the project, so the work that we're doing on long-read sequencing, we're hoping to use some of these algorithms and existing algorithms to see if we can improve the variant calling accuracy, especially for structural variation, because the typical NGS pipeline that is followed for rare disease is using NGS, and we know that many copy number variation and large structural variation are missed when we are dealing with when we are using short read sequencing. So this project we have started in collaboration with STAN. It has been a few months now, and there we are the vision is that we will be going for long read sequencing, and we will try to get T2T genomes and whatever it takes to get as high quality variant calling and other aspects of the pipeline to improve the diagnostic yield in the process. So with time, we know that more than half of the samples, they're not able to get a diagnosis. And we think that maybe if we do a more careful analysis through long read sequencing and better algorithms, we may be able to pinpoint or we may be able to find diagnosis for samples which are negative exome right now. Did I answer your question properly, or let me know if you have more comments?
So you're saying like currently they do exome mostly for most cases.
Yes.
And a lot of the samples, they are not able to arrive at a confirmed diagnosis.
Yes.
Okay. This is work with Ramesh Hariharan, I know?
Yes.
Okay.
So we will be taking those negative exome samples and we will be seeing what can we additionally do using long read sequencing and additional algorithms.
These negative ones, had we done whole genome, would we have arrived at an answer? Whole genome is obviously more expensive and more time consuming. And
yes, so that's a possibility. So that we're considering whether we may go for both whole genome as well as long read. So right now we're exploring both, but the idea is to just try whatever, either whole genome or long reads, and see what incremental gains we make with these two technologies in the diagnostic field.
Please, anybody, feel free to ask any question. There is no silly question, or it's rare to get somebody of Professor Chirag's level one-on-one. If you have any question, feel free to ask. Okay. Maybe not.
In the AI part that you were asking earlier, so there is work going on AI using ML algorithms for assembly as well. And some of the components where success has been achieved is also the error correction of long reads. So this is recent work where it has been shown that deep learning methods also do very well on the initial error correction of long reads while preserving the haplotype specific information. Because one can always overcorrect and lose the haplotype specific information that is needed to separate the paternal and maternal haplotypes. So that is one area that is gone. And second is also to simplify the graph. I was telling you that once the graph has been built, how to simplify the graph and how to predict what edges to take during the graph traversal. That problem actually is also being under active development on. So there is Vijay Sikhi's group in Singapore who is working on this problem, where they're using graph neural networks to see whether one can learn on some data sets where gold standard outputs are available and see whether they can apply to other species in terms of resolving these graphs and traversing these graphs.
And the point you made, that was very important and going to be very impactful in the times to come, that maybe human reference is already there, GRCH38, but as the diversity of genomic data, that is sequence data that is being captured grows, maybe we want an Indian human genome reference, right? And the input to that could be like a specialized form assembly where you know the GRCH38 as kind of the rough starting point, and you want to fine tune it to arrive at a Indian human reference, right? For example.
Yes. Or it could be just a single global pan-genome reference which has all the populations represented. Both could be done. I mean, there is also ongoing work on using these pan-genome graph references where you have multiple
references. Pan-genome reference will be really big and will have all kind of subgraphs inside it.
Yes.
Okay.
That is one algorithmic challenge that one has to deal with. Whereas working with directly Indian reference, maybe, technically it may be easier.
Okay.
But one can say that within Indians reference, we would want to have multiple ethnicity within India captured. So that again raises the question of what is the best way to represent the Indian reference genome itself, right? So that depending on
maybe we first assemble an Indian reference and then use that as the input to develop India specific reference, like sub-India specific reference genomes, right? Like a North Indian reference genome, South Indian, a Northeastern, a Western India.
Right, absolutely.
And I think with the cost of sequencing, like it's been faster than Moore's law, like it would be not hyperbolic to say that you might be sequenced every year, like in the next decade or so, everybody would be sequenced every year, right? For example, or
yes.
So the company I was referring to was Element Biosciences,
right?
Yeah, which is the competitor to Illumina.
Right.
students, others, please, I feel bad. I am hogging all the questions. Anybody else has any question related to sequencing or genome assembly? Okay. If not, then let's all thank Professor Chirag for an excellent talk. And we hope to host you at Ashoka for an in-person talk, perhaps. And yeah, thank you so much.
Thank you. Yeah, thank you for this opportunity.
So the final talk of the day of the workshop will be at 3:45 by Professor Durba Sengupta of NCL Pune. Please rejoin at 3:40 or 3:45 and see you for the final talk.
Hello.
Everybody on the call, I just wanted to know what part of, like which city are you from? I know, I think Wazir is from the Delhi area, but others, where are you from? You can type in the chat or unmute and say. I'm asking because tomorrow we have a in-person um, workshop on the Ashoka campus. Most of you would have seen the invite. So if you want to attend, you are welcome to attend. Okay, Glasgow, Palampur. Okay. Richa, Shubham.
Hi, this is Durga here. Can you hear me?
Welcome, Professor Sengupta.
Hi. Yeah. I was wondering if I could test.
I made you co-host.
Thank you so much. That would be great. Let me just try.
And
you
we'll
chat
start
with
around
others.
3:45.
Yeah, sure. But let me just try because last time I was doing this, I couldn't. Yeah, I had some issues.
You are not audible, Professor Durba.
You can't hear me? Hello, can you not hear me? Hi, can you hear me now?
Yeah, that's slightly better. Yeah.
Okay, then I had my headphones on. Maybe it's better I remove them. Or I will try again. Let's try.
I think now it's good. Yeah.
Is this okay? Still? I just changed to a microphone.
Could be a little louder, just a little bit louder, I think.
Okay.
This
Is
is
this better? Yeah, then I'll just hold the mic right there. This is better, right?
yeah, this is much better.
Okay, I will try to change my settings in the meantime and check everything.
And ma'am, can you share your slides so that we can do a quick check?
Yeah, I'll do that. First, let me just check the mic if that works, just to make sure that it is okay. Yeah, you can hear right now, fine.
Yeah, perfect.
Okay, I will just check my screen, my file share as well in a minute. Um, you can see my screen, right? And is it currently in the presentation mode?
Yes, we can see the title slide.
Okay,
Can you just go forward a couple of slides? Yeah, it's moving.
it's moving, right. Okay, so I'll just unshare for now and then share back. Is that okay?
Yeah. We'll start in about 10 minutes.
Okay, sounds good. Thanks a lot.
Thank you. So attendees, I was just saying tomorrow, those of you who are attending the hands-on session, it will be basically an introduction to basics of programming like Python and a very brief intro to AI in biology, like how to use AlphaFold in a Jupyter notebook. So, and I'll send out the details to those of you who have registered, but it will be from 10 to 5 on the Ashoka campus in Sonipat. And to come, you can, I'll send the details, but basically you can take the yellow line and come to Jahangirpuri Metro, and then we will try, I can't promise, we'll try. There is a shuttle, Ashoka shuttle which will take you from Jahangirpuri Metro to the campus and back in the evening. But yeah. Professor Durba, I think we have enough people. We can get started and maybe end 5 minutes early, if you're okay.
Oh, sure, of course. Good. I will reshare the screen
And
and go back to my presentation, which you can all see now again, right?
yeah. So I'll quickly introduce you and then we can get started.
Sure.
So welcome back everyone for the final talk of the workshop and the final talk of day 2. Thanks for sticking around, and we are delighted to have Professor Durba Sengupta of NCL Pune, National Chemical Lab Pune. So Professor Durba is a faculty member at the NCL Pune. She did her PhD from University of Heidelberg, Germany, in the group of Jeremy Smith, and her postdoc at University of Groningen, Netherlands, in the group of Sievert Jan Marink. Her work focuses on membrane protein dynamics and understanding self-assembly of biological systems. Her talk today is titled Multiscale Biomolecular Simulations. Ma'am, please let us know about Q&A. Would you welcome questions to your talk? Or...
Yes, so I think what I have planned is just the talk is in multiple parts, so basically 3 parts. And so at the end of each part, I'll anyways pause and you can ask, but you can also, they can also ask at any point, stop me and ask, because I wasn't sure of, the audience itself, how much of the background to give and how much to take for granted, especially because you've had a couple of MD talks. I know Lipi spoke, I know Anand spoke. I briefly heard them a little bit, but I didn't go much, didn't attend the whole thing. So if I'm saying things that you already know, you can, unmute and say, go fast. Or if you think I'm going too fast, they can also ask me to stop and slow down. You can ask me questions in the middle if it's super urgent. Otherwise, I'll anyways pause at the 3 parts separately. Is that okay? Does that work, Shish?
Yes. And I would just like to say to all the attendees, please engage, please ask questions. Otherwise, it's just a one-way monologue. I know it's after lunch and end of the day, so everybody's tired, but please engage. No question is too silly or too basic or too simple, or please don't hesitate at all. I think the speaker is also encouraged if it's more interactive. So please raise your hand and feel free to ask questions. Ma'am,
Yes.
please take it
Thank
away.
you so much. And I really appreciate you really stressing the point about questions, because I think that's part of the job of the workshop. Otherwise, it's really one-sided, and 1.5 hours is really a lot of time for me to talk into a wall, right? It's just a metal piece that I see in front of me, and I'm just talking to my metal laptop, right? So I'll really be happy if you all ask questions. So please, just for that, even if you think it's the stupidest question, please stop me and ask me, because I really appreciate that. Okay, thank you so much. I know I'm standing between you and the end of the workshop, so many of you just want to go home fast. So I'll try to make it as brief as succinct, but I've also kept it in 3 parts. So what I'll talk today about is basically looking at different scales in biomolecular simulations. And I purposefully didn't call it biological simulations, because you've actually seen many different scales over the course of the workshop. But even looking at molecular effects in biology can have actually many different parts to it and many different aspects to it. So I'm going to talk about 2-3 of those today and see how we can, pull in all of those to answer questions and see what we do with them. So in our group, we basically work with this system. This is the cell membrane. So that sits around every cell. And for everything, whether it's information, whether it's food, whether it's a drug, whatever it is, it needs to cross this barrier to go from outside the cell to inside the cell, right? And you can imagine if your laptop, your cell phone, your security guard, your fax machine, your Swiggy, your Zomato, everything had to go through this, right? Because it does get everything in. So all your posts, everything is going through this. So it's really quite complex and important. But today, I'm not necessarily going to be talking about the cell membrane. There'll be little bits of pieces because they are in all our work. But because it's so complicated, we realize that we often need to look above or below or further away from the cell membrane in order to truly understand the cell membrane. So what I'm going to do today is take up systems that you will see have multiple roles in the cell and try to correlate all that back in. But let's just still stick to the cell membrane for a minute, and let's try to look at least even at the cell membrane, what are the different processes that occur at it, right? So you have protein dynamics, you have lipid dynamics, you have protein-lipid crosstalk, but then you also have lipid diffusion, you have protein organization, and you have a whole range of different processes, right? And each of these processes, they occur at different time scales. So the fastest local protein dynamics would occur at picoseconds to nanoseconds, right? So of course, side chain movement happens at picoseconds, and that can be very important for certain signaling effects. But even slightly longer domain motion that occurs at nanoseconds is super critical for all kinds of signaling that occurs at the cell, right? So you want to go from nanoseconds to all the way to seconds, because two proteins need to crosstalk. Let's say you have protein association. They need to come together, they need to interact. And once they interact, only then a small helix moving away or inside actually makes the whole difference to the cell signaling, right? So you have this whole range of processes that occur at different spatiotemporal scales, so at different length scales, at different time scales. Now, if you want to study them experimentally, you would use different methods, and I've just put down a few down at the bottom, like FRED, FCS, NMR, and each of these experiments also occur at different time scales, right? So if you want to look at phenomena that happens at nanoseconds, you will do FRED for them. But if you want to look at phenomena that happens at milliseconds, you may try to do some kind of NMR over there, right? They are trying to do more and more in vivo NMR, right? If you do NMR at events that occur at nanoseconds, you're going to miss it. You're going to see just an average picture. Similarly, just like every experiment has an inherent time scale that you probe, you also have different computational methods that probe these different time scales. So you can't use a method that is suited for probing a different level of phenomena or a different level of process and use it to sample something just off the key, right? So there is really no one computational method that fits all, especially at the molecular level, right? So if you're looking at nanosecond level processes, and this could be protein dynamics or even lipid dynamics, you would look at what we call atomistic simulations, right? And if you want to look at things that happen much longer, you could use the same methodology, the framework, but do something that is called enhanced sampling, right? So what we do a lot of in our group is basically atomistic simulations and something that we call coarse-grained simulations. I think Shachi would have given you a little bit brief about that, but the rest of my talk, I'm going to go more into that. And in particular, the flavor of the coarse-grained simulations that we use is called Martini, right? So atomistic simulations basically consider every atom. That's why they're called atomistic. So every hydrogen, every oxygen, every nitrogen, everything is there considered in our systems, right? Now, coarse-grained decides that, well, you don't need everything, right? So you can kind of club things together. So you still have them implicitly present. So you would have a methyl group, or you would have a hydroxyl group. Sometimes you can even have an alkyl chain. And you may not be able to see the individual carbon and hydrogen, but you know that it is reminiscent of an alkyl chain. So I often think, and this is an analogy I often give, which is that atomistic simulations are like if you're wearing glasses and you see everything, or even, you have those fitted visions, you probably all have been seeing the shooters do it. You see things very clearly, you have a high resolution. But if you take off your glasses, you can still make out people, you can still see the blurry backgrounds, but you don't know exactly what is what, right? So you can see my bookshelf, but you can't see which book is there, right? But if I hadn't put it to blur, you would be able to see exactly every bit over there, right? So that's the kind of coarse graining that we are talking to. And there are multiple different flavors of coarse graining. I will not go into that today. I do teach a course on that often sometimes, but that's really a kind of a tangential thing. So I'm going to talk about things that we do in our group, and namely the Martini coarse grain model, right? So let's just look at what is first, what are these frameworks? What are we doing when we are doing atomistic simulations or coarse grain simulations, right? I think you all have a brief idea, but let's just recap. I think it's always good to recap before we start off, right? So if you have a single particle, right, I've just put one particle, and it can either be an atom or it can be a bead like we use in our coarse grain models. It's free to go anywhere and it can do anything, right? So you give it a force, it will go in that direction. If there's solvent around, it would have Brownian motion, or so on and so forth. But what if it's actually bonded to another atom, right? So now 1 is kind of dictated also by 2, right? And this interaction is given by what we call, we have very simply in molecular dynamic simulations not looked at the actual bond energetics, but just given a very simple harmonic function to deal with this, right? Similarly, if you have another atom which is associated with it, we have an angle which is also a harmonic potential. Then you go on to the dihedrals. Now 1 is dictated by 4, and how 4 behaves also dictates how 1 will move. And you can go along the chain. Now there'll be weaker and weaker coupling, but they may start interacting again through what we call non-bonded interactions, which could be electrostatics or van der Waals types, right? So basically, molecular dynamic simulations is just a mathematical framework which writes down all these different aspects and then basically solves the Newtonian equations of motion to get something like this, right? So you see your bonds are stretching, they're vibrating, your angles are vibrating, your dihedrals are rotating, and you will be able to get this very highly resolved, you know, movie of your biomolecule, if I may say so, when you do molecular dynamics, right? So basically, the force field is nothing but a summation of all these different terms I just explained. So what you get is a very, very high resolution movie. But from that movie, you still need to extract information. And to extract that information, we use statistical mechanics, and we consider each of these states that we get as microscopic states. We can then use StatMech to actually get ensemble properties and look at even, you know, collective behavior or collective observables that we can see then, such as pressure, density, temperature, and so on and so forth. So it's often not just enough to get a single movie. You have to make sense of that movie. You have to understand that these are single particle events that you're seeing. You need to get enough statistics to put all of this together, and you need to put it in a very robust StatMech framework to go from single particle positions that you see in the movie to actually making sense, right? So this is all great for atomistic simulations, and this is kind of easy to understand if you've had any thermodynamic class or something. You have an atom, you know what it's doing, you can understand it. But what we do when we do coarse grain is we kind of put everything together, right? So now, as I said, you no longer have atoms, but you have beads. So for instance, those of you who are familiar with proteins or amino acids in particular, let's say you have the small amino acids like glycine and alanine, they'll just be one bead. We don't care if it's a backbone, if it's a side chain, what it is. It's small enough, it's just one bead. But something which is very large an amino acid like tryptophan, we may consider that to be a set of 5 different beads, right? Because it has a much larger side chain. We have 1 bead for the backbone and, you know, 4 beads for the side chain. So whereas tryptophan actually has almost 40 to 50 atoms, if you consider all the hydrogens, if I remember correctly. And so we have just gone down an order of magnitude into just 4 or 5 beads, right? So now my calculations just have to be a lot smaller, right? I'm not calculating everything. So you can imagine that what I do now is much, much faster, right? And so this is really what is useful in biology. So if you're, you know, trying to compare your calculations to very detailed physics or chemistry problems, then probably atomistic simulations are the way to go. But in biology, we are often considering spatiotemporal regimes which are large, very large, and therefore in our group, we use this Martini framework a lot to try to understand that, okay? So you see that for a lot of things, we really simplify. Water is just simply a ball, right? It has no charges in our framework, right? And there are, of course, pros and cons associated with it. And unfortunately, because of that, we have to redo a lot of things, which I will be coming into, right? Because if you don't have hydrogens, how do you have hydrogen bonds, right? So that's, I think, the biggest thing that you need to consider, that if I don't have the hydrogens specifically put for my water, how will I have the hydrogen bonding capacity of water? And therefore, some of our biggest problems with looking at coarse-grained simulations for biology is handling water. You would imagine water is trivial and easy to do, but it's not, believe me. And so we do a lot of, roundabouts and fixes, and I'll show you some of the fixes that we are using in our group in order to do that, right? Lipids, which are the main constituents of the membranes, are on the other hand easier because they are just much longer. So you have a few beads that look at the acyl tail regions of the lipids, you have a few beads that look at the head regions of these lipids, and all of this we would just call the overall Martini framework, right? And each of these beads have a certain flavor. So they may be hydrophobic, they may be hydrophilic, they may be polar, they may be charged, but these different flavors are quite similar, okay? So I'm going to talk a lot about both atomistic simulations, well, actually, and much, much more about coarse-grained simulations. Start with is first try to convince you why I'm using this Martini framework, and then I will come to why not Martini and what else we can do, right? So let's start with why we use Martini. I've been saying this a lot. So I just put down some of the work that we have done in the past in different forums. So for instance, a group works extensively on protein cholesterol interactions, and in particular on GPCR cholesterol interactions. So we have really looked at how cholesterol binds a large number of membrane receptors. What I've shown here is actually the EGFR, or better known as, in this particular case, it's ERB2, or better known as HER2. So these are also very important receptors as well, and we have shown how cholesterol can actually modulate its structure and therefore its function. We've also looked at how membrane dynamics happens in membranes, and we have looked at large number of membrane peptide interactions, and in fact I will be talking about some of those work today, but we have looked at some other proteins, let's say caveolin, and looked at how that can interact with it. And for instance, we go from the membrane peptide interactions to looking at how then that interacts and gets curvature in the membrane. So we've done a lot of work on protein-protein interactions, protein-lipid interactions, membrane dynamics, and even we have tried to look towards ligand binding, right? You would imagine that ligands would be something that would require very specific hydrogen bonds, you would require very specific atomic detail, but a lot of times the ligand for many of these membrane receptors are basically also proteins, right? So like the chemokine receptors, which are GPCRs, their ligand is a chemokine, that's a protein. So when we look at these protein-protein interactions, we can actually say a lot about their mechanism. But however, I think this really forms the kind of, the good picture of Martini to say why we should be using Martini. And Martini has been used extensively. Recently, there was a review at looking at some of the best and the brightest or the biggest things. I think this one, longest single simulation time, that's us. So we have really pushed it. So even in a group which does not maybe have access to the best computers, let's say Anton, which is, or the, the fastest, the top 100 supercomputers in the world, we can still, using these coarse-grained models, do a lot, right? And in fact, funnily enough, this was one of the first work long ago I did after we started working at NCL. So this work was done actually 10 years earlier when we had even less computing time, right? So that's really the power of Martini, that we can really push it, okay? But what I'm going to talk about today is some system where we cannot push it, right? So I'll give you a bit of an overview, okay? So the main protagonist of today's talk is this protein, which is coffulin. And you'll keep hearing me saying the word coffulin again and again. So it's basically this protein down here. Now it's what we call a moonlighting protein, right? What does moonlighting mean? Basically, it has multiple jobs. So its job that started exciting us is that it goes and binds to the membrane, right? So we were very interested in how it binds to the membrane. But it also then actually just resides a lot of time in the cytoplasm. So it goes out of the membrane, goes into the cytoplasm, and then it actually goes and interacts with actin. So this is a very unique protein that interacts with another protein, that is the actin filament, as well as the membrane, right? And in doing so, it needs to keep shuttling between these two places. And they're quite different, right? The actin is a protein. It's in the cytoplasm, so it has a lot of water, like, it has to be in the cytoplasm. It is basically made of many different globular proteins, whereas membranes, they're very, very hydrophobic, right? So it needs to come and bind to this hydrophobic surface, and yet it needs to bind to the very charged actin filament, and it needs to be stable enough in water when it's shuttling between these two, right? So this is a protein which has like many different, function, of course, but it probably therefore has many different structural aspects to it, which allows it to go to all these different places, okay? So I'm going to talk about a bit of these. So the first part of my talk will be on how this protein binds the membrane. That's the most complete work that I'll be talking about, right? So that'll be the part 1 of my talk, and that will be focused on this Martini gauge-drain force field that I said. The second part will be how this cofulin actually resides when it's in water, right? So what does it do? What is the dynamics? Can we still use Martini? Because we have in our group so far only used it for membrane, so we were not sure. So I'm going to talk to you about different checks that you do, because you should not use anything just off the box. So you should actually check whether these parameters that you're using, whether the frameworks you're using are actually valid in this case. So I'm going to show you some validation of the Martini model itself, and that will be a little bit more didactic. And then the third part will be that what else can we do if we can't use Martini, or where can Martini work and where can it fail? And then I'll just have an epilogue on how it can actually go and interact at the actin, right? So these are the 3 parts of my talk today. And before I start off on the first part, which is the cofulin membrane interactions, maybe I can take a brief pause and see if there are any questions. Or maybe I can take a brief pause and force a few questions.
Please, anybody has any questions, please feel free to ask.
Yeah, no, it's also good to have all this info seep in. So I will not force a question, but if there is a question, that's good. And if there's no question, then you let that seep in. But basically, just to know where you're in the talk, you will be at these 3 points. There'll be the first part, which is on the membrane part, second part, which will be in water, and then the third part, well, I'll tell you what else we have been doing with this model, and then we'll be wrapping up. Okay? So if you're all on board, and if you're all with me so far, no questions, then let's go and start with the part 1, right? So let's just look at this protein. It has multiple alpha helices. It has all these beta sheets. It looks like any other protein, right? And when we started off, we kind of just started off, right? This was actually something that we didn't think a lot about when we started, which we should have. And partly because this protein looks very different than all the membrane receptors that we've used in our group so far, right? So we look at our group a lot at GPCRs, at other membrane proteins, EGFR, all kinds of receptors that occur at the membrane, right? Now, here was this protein.
I had a quick question.
Sure.
What is EGFR?
That's the erythropoietin growth factor receptor. So that's the one that is basically telling your
cancer.
Pardon?
Is that also implicated in cancer?
Yes, very much so. That's why I said also HER2, because cancer biologists always think of it as HER2, but it has many names. So it's either called ERB1 or EGFR, ERB2 or HER2, and so on and so forth. So because it's a growth factor, so it decides whether your cell will grow or not. You can imagine it's one of the main targets in cancer. So because it's growth, right? Cancer is just uncontrolled growth. And in fact, the cell has to decide metastasize or grow, and therefore this is one of the important targets. But I was just using it as an example to say that all of these reside in the membrane. And here we were working with something that does not reside in the membrane. Now, a priori, that seems very trivial, but actually it's quite different, because all these receptors that stay at the membrane, they're quite hydrophobic because they need to talk to the hydrophobic membrane. Whereas all the proteins that stay out of the membrane, they should be hydrophilic because they need to stay out of the membrane. Now here was a protein that had to do both. So it had to have enough hydrophobicity as well as enough non-hydrophobicity to be able to do both, right? So we started working with the system. we started thinking of what kind of membranes were we interested in. We looked at some complex membranes because this cofulin has been shown to be interested to bind the membrane only when there are PIP lipids, okay? So in fact, PIP2 has been really implicated in this particular cofulin protein and how it comes to the membrane. By the way, actually in biology, I think a lot of things are implicated in cancer. Even this protein is implicated in cancer because it's also important in metastasized cells, right? So basically when PIP2 is at the membrane, it kind of can't do its job at actin, but then it also bends membranes and so on and so forth. So it has just so many different roles that people aren't really very careful about it, or they don't know all about it, right? So it was always very, what should I say, very casually put in a lot of research articles that there is a role of PIP2 lipids in cofulin action, but it wasn't very clear. Now PIP2 is this lipid that I've drawn over there. It actually has a sugar moiety in its head group, which you see this 6-membered chair-like thing, which has lots of oxygens. And then on top of that, if there are 2 phosphate groups, it's called PIP2. If there's 1 phosphate group, it's called PIP1. If there are 3, it's called PIP3. And if there are none, it's just PI, right? So there is a whole range of these PIP lipids in the membrane. And usually they're very small in amount. So there is about 2% of these lipids in the membrane. Our majority of the lipids in our membrane are POPC lipids, right? And then depending on which cell type you are, the next is cholesterol. So if you're in your brain, you have about 40% cholesterol. If you're in your, any other general, let's say epithelial or cell, you will have about 30%. If you're down in certain places like gut, you'll have 20% cholesterol. But basically, there's a lot of cholesterol as well, right? So most of the people focus on the more important lipids used to, which were 40% of the membrane or 50% of the membrane, whereas PIP2s are only, or PIPs in general, are only 2%. And it's also very difficult in experiments to distinguish between PIP1, PIP2, PIP3. Of course, you can synthesize it and make it and put it, but in the cell, people are still trying to get good molecular sensors or good chemical sensors that can identify PIP2 versus PIP3 or so on and so forth. But what we do know is that these lipids are really critical in a large number of different functions, but often because they cannot distinguish between PIP1, PIP2, and PIP3, they just call them often just PIP2. They just say, okay, it's PIP2, and that's what we claim is important, even though they know that's not entirely true, right? So there was a role of PIP2, so we considered this very complex membrane, and we had to, right? Because we can't increase more than 2 to 4% PIP2s in the membrane, so we had to have everything else, which was quite correctly put. All the other lipids were in the correct ratios. And what we did was we placed this protein in water, we put it close to a membrane, and we did just out-of-the-box Martini simulations, right? So these are very simple, very straightforward coarse-grained molecular dynamic simulations, and what you see is in the simulations, you have your protein, it starts diffusing in the water, and at some point it comes and binds to the membrane. Over here, you can see this is the membrane, and you can see very clearly this red protein has come and bound to the membrane, right? So this is kind of out of the, just using simple coarse-grained simulations. And we were kind of happy because the protein did what it should do, which is combine the membrane, right? And then we started looking at how. So we first started quantifying what it is doing. So you see at the start of the simulations, you have all these, blue lines and red lines. So this, which signify the distance from the membrane. So red means far away. So this basically means your protein is diffusing all over. And at some point you see only dark blue lines, which is low distance. So it means the protein has come, bound the membrane. And once it binds the membrane towards the end of the simulation, it's all blue, meaning the protein is just bound to it, right? So now I have very nicely shown that this, in our simulation framework, this protein can come, it can bind the membrane, and it can stay on the membrane, right? And now we can start looking at which lipids are close to the protein, right? So we do it through something that we call occupancy. This is just based on the number of contacts. It's something we use in our group a lot. And you see, for instance, this DPCE, it's a ceramide lipid, is barely have any contacts with the protein, so it's quite less. And you see the maximum occupancies, this blue and magenta, are PIP2 and PIP3, they are quite high. But you also see POPC and POPE are there. But remember, there's almost 40 to 50% of POPC and 20% of POPE. So these making large contacts may not be that relevant, whereas these 2% lipids making contacts might be more relevant. So we started looking and seeing if these are clustering, and I'm not showing you all the data here, but you see very closely right in the middle there's a dark red spot, and that means these lipids are coming and they're kind of clustering. And that's not the case with the other lipids. But, and what was even more interesting for us is that if we just remove PIP2, once the protein has bound, you know, this is the first part till about 4.5 is where the protein is bound on the membrane, it's happy. We remove PIP2, and in one case, it just goes out of the membrane and starts diffusing everywhere, right? So you need to keep PIP2 in the membrane in order to keep the covalent on the membrane, right? So you saw this data before, the total occupancy. So just to confirm with the radial distributional functions that many of you are aware of, this would be the first peak. So this would be how much of lipids are in the first shell around the protein. And you again see this cyan and magenta are right up here, which are PIP2 and PIP3, and therefore they interact the most, right? And you see this black and red are down here, and that's because you're in an RDF, you're actually averaging over bulk. So because there's a lot, you don't see as much, right? But we were very puzzled why PIP2 and PIP3 were showing up all the time. So sure, PIP2 is a bit more, but PIP3 is also less. And we actually went back to the original experimental papers, and we saw that even they saw PIP3, but they haven't mentioned it anywhere. This is in a supplementary figure hidden somewhere away, right? So they keep on saying everywhere in the title, in the abstract, in the main text of the whole article that it's always PIP2. But if you see that paper, sure, in the supplementary you have PIP3, even though sure the deviations are very high, but even PIP3 does have an effect in actually determining the binding to the membrane, right? So our simulations were kind of able to pick up that PIP3 is actually also good to go. And even if there's, you know, it kind of interacts with the protein and stabilizes it once it binds the membrane, okay? So you can see now very clearly where does the PIP2 bind, where does PIP3 bind. So you see PIP2 has this huge interface. These are the actual numbers, and we have just mapped it on the structure. And you see there are lots of these blue spots. That's a lot of charged, positively charged amino acids, in particular the lysines. And you see that PIP2 forms a very nice long continuous line, and PIP3 and PIP1 are less continuous, but still very much there. And this is why once the protein binds to the membrane, it kind of starts to cluster all these lipids together with it, right? So because naturally there are many binding sites of these lipids on the protein. So once it's at the membrane, each of these come together. And this very unique set of residues, right? Because there are a lot of these charged lysine residues, they kind of have the differential interactions with the PIP2 and PIP3, right? So we next wanted to ask, I mean, what is, you know, why is both PIP2, I mean, what is PIP2 and PIP3 doing? And in particular, we wanted to ask ourselves if there is any kind of, you know, how are they actually binding with each other? Wait, I think I went ahead, right? So we went back to trying to understand how can we quantify whether these lipids bind together or they bind one at a time, because obviously they're binding a lot. So we use something called the Hammond coefficient, or also we looked at similar coefficients like, you know, we picked it up from a very different profile. So it was actually proposed in the early 1900s for ecology. So they wanted to know, using these coefficients, whether, let's say, two trees bind in the same ecological region, right? So they wanted to know whether they are synergistic in their growing, the trees, or they're competitive in their growing, or they independently grow in different ecological environments. However, these days, this is really used most importantly for all your online shopping purposes, right? So when you buy something from Amazon, it always gives you a suggestion. People who bought this also bought this, right? So they actually use these coefficients to quantify whether buying a certain thing is in synergy with what you just bought, or it's in competition. So if you bought, you know, thing A, how likely are you to buy thing B, right? So we wanted to use a very similar framework to come to whether we can actually quantify this PIP2 and PIP3, you know, competition. But basically, we saw that they were weakly competitive, right? So they were either independent or weakly competitive. And that's probably because there's no one binding site. If there was a binding site, they could compete for the same binding site. But what they were doing was they were just clustering all the lipids. There's a very extensive interface. They're looking at all of these. And so it doesn't really matter how they are doing that, right? So if PIP3 is there, PIP2 can also be there. So it's kind of independent binding because it's such a large interface, okay? So I think this whole part of the PIP2 binding went off really well. We were even able to see how the PIP2 specificity is coming, whether it's the charge or whether it's the geometry. And this was really surprising for us because coarse-grained models are not really able to give that much resolution in terms of either charge or geometry. So we did what a lot of people do, which are mutations, but people mutate a protein residue into another. What we mutated was one lipid into another. And that's very simple to do in our computational frameworks. You just take PIP2, add an extra phosphate, it becomes PIP3. You remove an extra phosphate, it becomes PIP1, right? So we could make our PIP2 appear like PIP1 or appear like PIP3. We could also just not make it actually PIP3 or PIP1. We could just change the charge on it. So it's still a PIP2, but having more charge, or still a PIP1, but having less charge, and so on and so forth. So after all these alchemical transformations, we could basically say it was both charge and geometry that was important for PIP binding, right? For covalent binding to these PIPs in the membrane, okay? So what we then thought was that, okay, it's great. We know now that it can bind to the membrane. We know that what causes it to bind, and we know what will cause it to unbind as well, right? So given that, can we now look at the next part of my question, which is once it unbinds, what does it do, right? So now remember, when it was bound, all these charged residues were looking at these charged lipids and keeping the hydrophobic surface, right? Now when it's in water, both the hydrophobic part and the hydrophilic part is kind of exposed. So the protein dynamics will be kind of different, right? So what we wanted to do is actually look at covalent in water. But there were many issues that we had to consider over here, okay? Before I go into all the issues that we considered, I'll take a brief pause and see if there are any questions on this part so far, which was on the covalent binding to the membrane using just basic out-of-the-box Martini simulations. So all of you are still with me, right?
Yes.
Well, you are, I know. I don't know if the others are.
People, please interrupt and ask questions or suggestions.
Yeah, no, that's difficult to force people to do, I agree. But it would be great if somebody could just say, hey, I'm here. Okay, I will just assume that all of you are still with me, and we'll go on to the second part, which will be a little bit more didactic. I think the first part was a bit more biological, trying to ask and answer a biological question on these membrane protein interactions. From now on, it will be a lot about comparing the force field, comparing the framework, improving the framework. So there'll be a lot more methodological part. So I hope you're all with me so far. Okay, so now let's go to the second part. So when we started looking at covalent in water, we started having problems of structural restraints, right? What do I mean by structural restraints? You all know protein structure is made of, let's say, alpha helices or beta sheets, but at their heart, they're just hydrogen bonds that keep an alpha helix stable or a hydrogen bond that keeps two beta sheets stable, right? Now, if you don't have hydrogens, as I said, we don't in our coarse-grained simulation, how do we have hydrogen bonds? So you kind of predetermine that this will be an alpha helix, this will be a beta sheet, and incorporate that into your structure. Now that's not into your mathematical framework. Now that's not very difficult to do because we have a whole PDB with lots and lots of structures, and we of course had structures of covalent. So we know a priori what does covalent structure look like, right? So that was not that difficult to do. But then there are different kinds of structural restraints. I think Shachi spoke a lot about these Go models, and that's definitely one framework. But you could just put a harmonic restraint there as well, and people have done that. So we were not sure We knew exactly how to handle the membrane proteins. We weren't very sure how to handle these structural restraints in water, right? Because the dynamics of each of these secondary structural elements will be different. So in particular, which exact Martini model to use, and when we started off, there was a new Martini model that was published that was called Martini 3. We were very used to Martini 2. Now, normally you would just say use the latest model, but that's not as simple to do, as I will explain over time. So we had this question, what Martini model to use? Electrostatics seem to be getting more and more important, and therefore there is always a problem with electrostatics, and that can be seen very clearly here. So what I've marked is just in these different models, certain salt bridges, okay? It doesn't matter which salt bridges they are. They're just two charged residues far away, close by each other, and I've just calculated what is the Coulombic interaction between them. So these very long bars that you see, they're always, the first ones are always atomistic, they are very, very long, and you see the other bars are all small, and the other bars are all the different Martini models. So basically Martini can't really ever come close to atomistic models in terms of how they deal with electrostatics. Now that could be a problem. In membranes, we know that's not a problem because I specifically showed you this pit lipid case where it's not a problem, right? So if that's not the case, then how does it work in water? So the next part of my talk, the part 2 of my talk, is going to be talking about how good is this Martini model, and we are going to be talking about two particular versions, the best two versions. All the bad versions I've skipped out from the talk just for sake of time, and we are going to be talking about this Martini 2 and Martini 3 and just comparing between them to see which is better, which is worse, okay? And what I'm going to do is I'm going to compare a little bit to atomistic work, but also more importantly to experimental data like SACS or NMR, because at the end of the day, that's the true gold standard in biomolecular simulations, okay? So this is my covalent protein. Now I have stripped off the membrane. I'll be talking about, for the rest of this talk, only about covalent in water, not considering the membrane, right? So this is what it is. But because we wanted to do a comprehensive testing of what is a good model, a good model cannot be dependent on the protein you're doing. A good model has to be general. And so we also considered other proteins in water to see if there's any general principle about choosing a the exact flavor of the models that we use, right? So we have these other proteins, as I said a lot about covalent, but we used all these model proteins that simply just used as model proteins. We don't care about their biology. We just care to see if we can reproduce how they work in simulations. And we chose these because a lot of people over decades have been doing simulations and experiments of these two proteins, and there's a lot of data on these two proteins. So we first wanted to see if we can reproduce the data that is already there on ubiquitin lysozyme, of course the one that we are most interested in, covalent, and that will help us understand what framework to use, right? So the first thing that you can do is something everybody who uses molecular dynamics does, which is RMSF. That's like the first student level thing that you do. And you see in the three systems and the three models that I have, the black, the atomistic, the green, the Martini 2, and the yellow or orangish yellow, the Martini 3, they look identical. They're on top of each other, and that's great news. And there are some differences, but let's not go into that. You can then calculate what is called protein-protein contacts. So everywhere there's a red line, red dot, it means two residues of the protein are close to each other, right? So naturally the diagonal is red because everything is close to the next residue, right? So before and after, that's why the diagonal is red. But all these lines that you see coming off the diagonal, those are basically the secondary structural elements, and everything you see which is completely off the diagonal, those are all the far off tertiary contacts. And basically what you need to understand is all three, atomistic, Martini 2, Martini 3, they look similar. So there is a lot of similarity between these models with the atomistic model, okay? So we said, great, we can do as well as atomistic simulations. But can we do as well as experiments? So one of the experiments that we chose are SACS profiles. So this is small angle X-ray scattering. So unlike the X-ray scattering that most of you are familiar with, where you get distinct diffraction patterns, you get a SACS profile that looks like what you see on the right. But you can see a lot from these points. You can talk about protein shape and size, and in particular, you can come up with something which is called the radius of gyration, right? So from these Sachs profiles, you can estimate how big is my protein, roughly, right? So what we will try to do is we will try to take the experimental Sachs profiles, predict the Sachs profiles from our Martini simulations and see how well they do. Actually, they do really well, right? So the first, we had the data only for lysozyme and ubiquitin, not cofulin, and you see that we can really come up with this quite well, which is kind of surprising because we don't really, we impose a structure, we do certain things, and then we can actually, from the coarse-grained model, really say that. What was surprising though is that the radius of gyration, which is the exact size, did not really match. It matched better with this last thing here, this Martini to go, which was actually the worst of our models, okay? So sometimes the worst model just happens to give you a good answer for all the wrong reasons. And I just kept it here to show that even though the experimental values are up here, 1.5 in this case and 1.3 in this case, most of our good models, including the atomistic models, lie well below that. And that's because in experiments, you also have a water shell that you can get along with the protein size. So you get the first water shell size as well. So the fact that these are actually match better, the ones which are the outliers, actually means it's actually the protein is too big. So it's actually doing worse, even though just superficially they seem to do better. That doesn't matter, right? We also looked at a lot of NMR data. So one of the things that we looked at is chemical shift. So basically where that comes in the NMR spectra. So what we do is we take our coarse-grained data. Of course, we need to have atoms to get NMR chemical shifts. We go from coarse-grained to atomistic and then use some machine learning frameworks not developed by us to come up with these kind of chemical shifts. And really, they do really well for both C alpha, H alpha, what have you, So I think the chemical shift data really matched well with our Martini data. But where we really came into problems was this J coupling, which gives these splittings, which you saw even earlier, but we kind of estimated what we call the calculus equation, and you can just kind of semi-guess what the values will be. And when we do that for our Martini simulation, so we go coarse-grained, atomistic, J coupling, they're just really terrible. They're all over the place, right? And so we did get slightly better ways if we do this back mapping slowly versus not so slowly and so on and so forth. But basically, J coupling is at the heart of secondary structure. This J coupling was how people guessed protein structures before they had X-ray crystallography so much in use or cryo-EM, right? So J coupling kind of really is very sensitive to secondary structure, and it's not a surprise that our coarse-grained models are doing so badly for secondary structure because, as I told you, it's not inherently possible in a coarse-grained model to have secondary structure, right? But for cofulin, when it comes into the water and when it binds into the backbone, into the actin, there are backbone changes that occur. So we would like to have this J coupling better, right? So the third part of my talk is going to be telling you, since Martini can't really model protein backbone dynamics, what can we do, okay? Let me take another pause. And if there are no more questions, I will drink water. And if there are no more questions, we will move on to the third part of the talk.
I had a kind of naive, silly question.
Sure.
I was wondering if you want to, like these, this modeling we are doing is like, why are we doing it? And like, does it make sense to do its interactions with blood plasma also, perhaps?
Not really, because these are all kind of within the cell. So everything that they're doing is shuttling within the cell at different locations within the cell, okay? So they are like busybodies of the cell, and because the cell has so many different kind of environments, so they're shuttling between all these different rooms of the cell, right? The membrane, where it's very hydrophobic. So it's like if you had a house which was very cold and you had in one room and very hot in another room, and flood in one room or fire in another room, you would have to have different kinds of shields to enter these rooms, right? So we want to know what are the shields of this protein that helps it go to all these different rooms at the same time. But outside, it's not really stepping outside the house right now to do other things. That will happen in a larger context in some other cases where you look at how cells migrate within that. But other than that, a priori, I can't think of a good reason to do that first.
And like, is it fair to assume that we are making an assumption that the medium in the cell can be approximated by water? Like, why are we using water?
Yes, so the medium is, of course, the cytoplasm.
Plasm, right.
Yeah, so I mean, of course, there's the membrane where we are at the membrane, right, which is a very different medium. The environment is just all lipids, which we do model. The cytoplasm, yeah, should have slightly different cases, but it does have a lot of water. And the point is that all the in vitro experiments that we can compare to, so all structural in vitro experiments are all done in buffer. So when we say water, it does have ionic strength. There is some kind of a buffer, but it's mainly water molecules. But we also have salts and other metabolites which may be needed to stabilize it. But those are really not the main aim, because what we see is that in our microsystems, these don't matter so much. So, but we do have, it's not really just water that we have, it's a buffer, but much, much simplified buffer with just some salt, which we have there.
Thank you.
Okay, thanks. So let's go on to the last part of the talk, which is that can we combine these atomistic simulations, the accuracy of the atomistic simulations with the speed of the coarse-grained simulations? Because while we were really getting interesting data out on the membranes, where we had these very complex lipids and it was kind of hydrophobic, we realized that we can't really get as much accuracy in just the coarse-grained framework, right? But coarse-grained models are indeed much faster. So can we kind of combine them? What I will say is that this is still work in progress. So these are methods that we are developing in the group right now. We don't have the perfect solution yet, but I would like to introduce to you one of the solutions that we are developing in the group, and that's the hybrid models, right? So you can do stepping between atomistic and coarse-grained in many ways. One is sequential. That's what we did earlier when we were going to look at J-coupling or chemical shift, because we took our coarse-grained model, we said for every coarse-grained structure, we went to an atomistic structure and then calculated all the properties. So you can keep doing this, right? You can start with a coarse-grained protein, do atomistic, or vice versa. And these are called sequential, but they're not really true multi-scale models, right? Then you have adaptive models, which are true multi-scale models, where depending on the space or the place where a certain protein is, it may be, coarse-grained or atomistic. What I'll be talking to you about is some hybrid models, where basically it is pre-decided whether your molecule will be hydrophobic, will be atomistic or coarse-grained, right? So let's say this is an example of one of the papers where they looked at these hybrid models for membranes. So you have certain lipids that are always atomistic, there are certain lipids that are always coarse-grained, and then you see how they interact over the time, right? Now this is not maybe as useful for lipids, but it's very useful for proteins. So I can have now my protein in atomistic, which is covalent, and I can have my solvent in coarse-grained, right? So I can keep the protein, which is the important part, right? And, and keep all the solvent around it. Okay. Now, if I do that, that's great. Atomistic models are very well parameterized to talk to atomistic models. Coarse-grained models are very well parameterized to talk to coarse-grained models. But we need to talk to these two together. So we introduced something called virtual sites. We could have, of course, reparameterized everything. And I'll come maybe in the discussion why we don't reparameterize everything. But we use something called a virtual site, right? So at every step with the atomistic model, there's this whole ball bead, which is actually virtual, which moves with it. So it's like if you tag 4 people who are friends all the time together, you don't need to look at where each of them are going because they'll all move together anyway. And so you can just tag that group of people as one, right? So you tag a group of atoms, which is exactly what you do in coarse grain, to be what we call a virtual center, right? And these are already quite well implemented in simulations. The virtual center, not this way of doing hybrid models, right? So AA, which is atomistic, talks to atomistic, coarse grain talks to coarse grain, but coarse grain talks to atomistic only via this virtual site, right? So in this virtual site, what happens is that you can all still hear me, right? I think I lost my cursor. Anyway.
Yes, yes.
Yeah, perfect. So in this, what happens is you have an amino acid. Let's say this is the whole atomistic region. You just put beads or balls, which would be the coarse grain. Now you know what this ball would act like. So it's called a virtual center. And then that virtual center talks to the coarse grain, whereas within them, they're free to interact by themselves, right? So certain molecules, and in this case, the protein will have both an atomistic and a coarse grain. And the coarse grain is actually just a virtual site implementation. I know it's a little bit difficult to get to understand, but basically now you don't have to reparameterize anything, but you do need to check your dielectric, how these virtual sites will be placed or mapped. So we had to do a lot of testing of these models to see. We had to get counter ions, as you said, for the ionic strength. It couldn't just be water. We still don't know what to do about electrostatics, but we are working on that, right? Now, these are plots you saw earlier. So these are, again, my 3 proteins. The ones below are the atomistic, which is what it would look if they are just atomistic proteins over there. And now what you had seen earlier were the Martini models, but now what you see are the hybrid models. So this is our model where we have an atomistic protein and Martini water, right? And you see that they're still doing quite well. So there is a good correspondence to the panels on top with the panels at the bottom. They're not perfect correspondence, but they're pretty good correspondence. So my protein is happy even in coarse grained water, which is kind of surprising because coarse grained water is not really a very good water model, right? Then we tried to fit back the SACS data. So these were again plots that you had seen earlier, and that's why I spent some time comparing so that you already are introduced to all these terms. So the first plots are what you had seen with the atomistic one. So you see that they really fit well, and a hybrid model does as well. And the same SACS data you can replot as a log-log plot or a log's normal square plot. These have special names in the SACS community, like the Greener models or the Kratky models, and those really fall well in our hybrid models, right? So the hybrid model is able to keep protein dynamics well enough, protein structure well enough. It can reproduce these experimental data. It does, again, really well also on the chemical shifts, but then the coarse grain models did as well. And our J coupling is really much better now, right? So we do have much better correspondence now with these hybrid models as we were having with the coarse, like much better than the coarse grain models, right? But even though now we have improved a lot of things and we are making still more improvements as we speak, we can already start looking at what I had said we will be looking at. So this is the epilogue of my talk. So basically what you see on the left is cofalin in pink and actin in blue. And you start seeing now, you can start looking at bigger and bigger complexes. So while cofalin itself is a small complex, you have this larger complex. And even this is not large enough for us. Actually, we need to start looking at even bigger complexes. So what you see on the right is an actin filament, which is decorated by multiple cofalins. And this is what we need to be simulating. So we have already started looking at these systems in hybrid, especially the dimers. And we can already see that the dimer is stable in our hybrid simulations, and it can kind of, reproduce what all it did in the atomistic system, right? But is that enough? So did we gain anything? The answer is not really. I mean, well, there is. It depends on who you're asking. Now, if you do atomistic molecular dynamic simulations on a normal cluster, not a like a supercomputer, sure, but it's still not, let's say, a mega cluster. Let's say you get about 20 nanoseconds a day for a larger system. In the hybrid, you're twice as fast, right? So you get 40 nanoseconds a day. But the coarse grain was always 1 microsecond a day. So coarse grain is always 1 order of magnitude more. The hybrid seems to do double as well as the atomistic, which is great because a lot of times atomistic molecular dynamic simulation takes a month, right? So now instead of spending a month to do the simulation, you get the same data in 15 days, okay? I did not really go into the details at all about the electrostatics. So that's still a problem, and I don't think this is the forum to discuss all those details. So basically, we still have a long way to go. Although we have a lot of improvements in the model, we still need to do a lot more to do that, okay? So let me pause here again. If there are any more questions on the hybrid side of it.
Ma'am, there was one question in the chat. Giri, do you want to go ahead and ask your question? Giri Lakshman?
Ma'am.
Please go ahead, Giri.
Before thinking about hybrid models, is there
Can
any
you
criteria
speak up, Giri? You are not very audible. Can you speak up a bit?
Ma'am, before thinking about hybrid models, is there any criteria to choose Martini method 1, 2, or 3?
Yeah, so that's why we did all the comparisons, because that was the same question we kept asking. But we saw that for most of the things that I showed you, both models that I showed, Martini 2 or 3, are equivalent, and we are finding out more and more. So in fact, a lot of the community claim 2 is better, but at least for the systems that I showed you, which is globular proteins, Martini 3 performs as well as Martini 2. Of course, the Martini developers would like you to use the latest, which is Martini 3, but we see that there are still issues with that. So both 2 and 3, it's a toss of a coin, or you do both and you see which one works better. So people are still doing that as we speak. But at least for the questions that I showed you, they're equivalent for the kind of tests that we did. And which is why the hybrid models I showed you are with Martini 2 right now. Actually, Martini 2 does better with water than Martini 3, but that's again a discussion for another day. And we are doing the hybrid models with Martini 3 as well.
Is there any advantage over 3, 2 over 3?
Is there any advantage over? Sorry, I couldn't hear you. Could you repeat?
What are the advantages of 3 over 2?
The advantages of 3 over 2. So 3 has been actually quite well parameterized, at least on the protein side. That's what they claim. Okay. So water dynamics, not water dynamics is something we tested. That's never been tested before. But protein dynamics itself is better in Martini 3 in their opinion. There are marginal differences, but not really a lot, right? It has been reparameterized to use the Go models. I think Shachi spoke a lot about the Go models as opposed to much more rigid structural constraints. But as I said, I think the whole community is still testing these models and deciding what is better. So if you want to use these models, I would recommend you to test both 2 and 3. Reviewers will tell you, hey, why didn't you choose Martini 3 if you choose Martini 2? So please do test out with Martini 3 as well.
Yes, some advantages of hybrid models over Martini. What are the advantages?
So the biggest advantage of the hybrid model, and that's something we are developing, so you should never ask a developer the advantage, because developers always say it's the best thing. You should always ask a critique, what is the advantage? You should ask a developer, what are the limitations? Yeah, so for us, the biggest advantage is that we can look at protein folding, unfolding, or protein backbone dynamics, because coarse grain cannot, Martini coarse grain cannot look at protein folding. So if you want to look at protein folding, that's not the coarse grain model to use. There are other models to look at protein folding, right? Whereas now we can actually look at protein folding and unfolding. And the reason why that was very important to us is that you have this protein called coffulin, which has slightly different structural ensembles in the membrane, in water, and at actin, right? So there needs to be some secondary structural or tertiary structural rearrangements that can go between these three, right? Now, hybrid therefore can do a lot more than coarse grain. But right now, hybrid cannot do a lot more than atomistic. The only thing it has advantage over atomistic right now is in terms of time, right? So you can just simulate a little bit faster. So it can give you very fast sampling, because for a lot of these systems, what we need to do is sample many different structural states. So unless you sample all structural states, you can't just say this is the best state. And a lot of people do that, by the way, with atomistic models. They just see something and they say this is great, but that's not how robustness works. So if you want to sample many things and then say this is great, you need to be able to sample all those. So if you can sample fast, that would be great. In fact, we are in talks with some of our colleagues to use these hybrid models to look when they are really looking at pathways or quantitative analysis where they really need to sample everything, they can now just sample it faster. So that's an advantage for them.
Whether hybrid models address the limitations of Martini models?
Does the hybrid model address the limitations of the Martini model? Yes, that's why we are doing it, yes.
What are those limitations?
As I said, protein folding, unfolding, or structural dynamics, right? So it cannot give backbone dynamics. That's as simple as that. Whereas my hybrid model can, because it is at the heart of it, an atomistic model.
Thank you, ma'am.
Okay, that's great. Thank you for all these questions. So yeah, basically then let me recap for everyone what I've been talking about so far. So I wanted to say first today, one of the take-home messages is that we do a lot of testing with different frameworks, different modeling approaches, and we usually use systems which are very well characterized, right? You either use globular proteins, and I know you'll be doing an AlphaFold workshop. AlphaFold works great with globular proteins, you know. That's because there's just so much data on globular proteins that it's been trained for, right? And then there are all the membrane proteins where it is now being training, so it does well on, maybe well on some membrane proteins that it's been trained on, or protein classes, so it does quite well for the core of the membrane protein, right? But now you have proteins that do both, and these should be the new frontier for testing these multi-scale models, right? So if you want a model that does, you know, is able to capture both the inherent environment of the membrane as well as the inherent environment in the cytoplasm, that would be something that should be the next frontier for testing. So I introduced you to one moonlighting protein, but there are many. We know that many proteins in the body have multiple functions, and we know this more and more, right? So these should be really what we target as our next test systems. I introduced to you the coarse-grained Martini model, and I said that it can model membrane process really, really well because membranes are kind of rigid and they impose rigid structural restraints. But I also told you, and we discussed, that they need to be really rigorously tested for water, so globular proteins basically. And finally, I introduced this hybrid model, which was used to bridge atomistic and coarse-grained resolution. Well, we have to test these models over long time scales. We need to extend this framework to much more complex systems. I've only given you very brief examples of where we can apply these to, but we have to use these for much larger systems as well, okay? So before I stop, I would like to thank all the people involved. The work was started off with Shikha, who introduced me to the Coughlin systems, and then taken over by Shalmali and Manjul in the group, who have been working on both testing the coarse-grained frameworks and building the hybrid models. Okay, so thank you very much, and I will leave you with this thought. Thanks.
Thank you, Professor Durba. By the
way, my name is Durba. Huh? Just you can check out all the ma'ams and professors and doctors. I should have said this right at the beginning. Sorry.
No. So thank you for a great talk. Any last questions before we end and have the final vote of thanks? I think they were in the chat. Hema, do you have any questions or clarifications? Okay.
Yeah, I think it's been a lot of info overload over the last 2 days and
Yeah.
at multiple levels and multiple things. So I think they're all processing the info still. So yeah.
Thank you. I am absolutely, I don't even have a biology background. I find that MD simulations to be a lot of math and stuff which usually just flies over my head, but your talk was very accessible even to somebody like me. So thank you for a great talk. And if there are no questions, then I'll invite the PI of the center, Professor Gautam Menon, to kind of give the vote of thanks and conclude.
You may have to unmute me and start my video, which I can't operate. Can you hear me?
Can you try now, sir?
Yeah, that might work. So thanks, Durba. I'm sorry I missed many of the talks in this session because I've been dashing between meetings from one place to the other. So I will have to save all of this up for...
Yeah, we should meet.
Watch it again online after that.
But you know, that's the problem with these online meetings, that you're not forced in one place. So we all, I mean, I've been trying to catch them up on YouTube, but failing at multiple places.
I think I will have to do that because I've really I've been watching your stuff and I would like to sort of see how you give the full lecture with all of the background inside it. It's been a while since we met.
Yeah.
But let me just use this occasion to thank not just you, but all of the speakers in this session, all of the speakers from yesterday. And it's been wonderful. Every time we do this, it's a way of bringing the community together, community of people who work on various aspects of computational biology, whether it be membranes or virus structure or AI and ML in novel protein design. And that's what I think is special about this. And there is an advantage, of course, to being online completely. It's easier for people from wherever, anywhere across India and any number to actually come and listen to this. And of course, all of these are resources that will be available online for people to look at. And as I said, the talks from earlier years are also available for anyone who wants to come and look at this. So I can encourage those who are listening to this, who on this audience, to tell your friends about it, to come back, look at the resources that the center provides, the Center for Computational Biology and Bioinformatics. And we hope to see all of you physically here sometime. I know that Ashish is planning tomorrow as a hands-on session, which maybe he's spoken about already, or can talk about a little after I finish. The person to thank really, of course, is Ashish, who put this program together, and we were discussing with both me and with Alok Bhattacharya. But all of the follow-up, recordings, interactions with all of you, etc., really completely Ashish's job. And I must thank him very sincerely for that. I'm very grateful for that. I hope all of you, those of you who have not visited Ashoka, can come here again at some point. And I wish all of you good luck and a great evening ahead of you. Back to you, Ashish.
Thank you, Professor Menon, all your mentorship. And I would just like to conclude by saying thank you for staying. I think attending 4 talks in a day with just 1 hour break can take its toll, especially in the online medium. It's much easier when you are physically in the same room. So, but we had a fantastic lineup of speakers. I wish more people could have joined, but feel free to, as Professor Menon said, share it with your colleagues, with your friends who are students or studying, or even faculty. These are some of the best folks in the country, or perhaps even in the world, in their specialized domains. So feel free to share these resources. And finally, tomorrow we have a hands-on session. So those of you in the Delhi NCR area, I'll send out an email with all the details, and most probably we'll try our best to arrange transport from Jahangirpuri metro station on the yellow line to Ashoka campus and back. And there will be some refreshments and lunch as well for those of you who can come. And next year or this year later, we intend to do a physical, like a conference style or a workshop style in-person session on the Ashoka campus. And I hope we'll send that out to everybody who registered for this. And we hope some of you can make it on campus at Ashoka as well. So with that, I thank all of you again, and I'll conclude.